All right, I think we're good to go. Okay. So basically, I'm going to be presenting today on abiogenesis, and I study biology, and I've always been interested in this topic. When I was in 5th grade, I asked my 5th grade teacher, how did life begin? And she told me, oh, molecules wanted to create other molecules. So I was like, what? And then ever since then, I've been really interested in the topic. And yeah, I've been studying it for about 4 or 5 years now and compiling data. So does science suggest abiogenesis or intelligent design? All right, so biology is the study of life and living organisms. Cell biology is the study of cells in particular, which is what I study. And you can see right here, it's kind of at the fundamental center of all these other fields of biology. And cells are the basic and fundamental unit of life. Fun fact, the word cell means cellula, which is derived from a small room. So a cell is just a small room. Came from that. All right, so fundamentals of biology. I'm going to start by running you guys through a bunch of fundamental concepts, just so whenever I make a point on biology, you'll understand what it is. I'm going to give you some information on the cell and how it works and the central dogma, et cetera, et cetera. All right, so basically everything's made of atoms. They'll make up molecules, which make up these organelles inside the cell. Cells will make up tissue, organs, organisms, et cetera, all the way until you get to Earth. And characteristics of life are they are made of one or more cells, they have organization, they can grow and develop, they can reproduce, respond to stimuli, they need energy, and they can maintain homeostasis or equilibrium. All right, single-celled organisms are small yet very amazing. So over here is a dancing diatom called Bacillera paxillifera, and here's a picture of them that I took on the bottom. So this is just a little cool video I threw in here. And here are some diatoms that are arranged in a cool, unique fashion by Klaus Kemp. And yes, these are living organisms, and they look interesting because their cell wall is primarily made of cilia. And then over here is another little protist. So they're very complex and intelligent and amazing.
Didn't we find out like these little
Yeah.
These little things are pumping back and forth, so they're like scooping up water. You know, like this is our digestive system in the middle, and they're just like going back and forth, just like pick up stuff to eat. I thought that was pretty cool. You know,
really interesting. All right, single cells, small yet amazing. Over here on the right, here's a cool little video of neurons. This little guy at the top's like over here trying to make some connections. We have a human cell reproducing. In the middle is the chromatin, and they're being pulled by the microtubules, which we're going to look at a little bit later. And then we have a neutrophil, or a white blood cell, that's basically chasing down a bacteria. So yeah, here's some cool videos of like what we look at in biology, some microscope videos, etc. And then here's what I study, microtubules. They are the cytoskeleton of the cell. They provide shape, structure, movement, and sensory input to the cell. And a little fun fact about these guys is they are attributed to fiber optic cables in the neurons. So that's pretty interesting. And here's a video of it happening, like an animation, and then a video that I recorded of the microtubules. And basically I was able to track these because these microtubules have a fluorescent protein at the end of them, so I can track them growing. So yep. All right, and then now we're going to move to intercellular components, organelles. So they have a lot of them. They have the cytoskeleton, which is what we just looked at, different types of them, the microtubules, etc. They have the cytoplasm, which kind of fills up the inner cell. Let's see, chromatin in the nucleus. It holds like the DNA. And then you have the endoplasmic reticulum, rough ER. This will make proteins, and then smooth ER makes lipids and stuff like that. You also have the mitochondria right here, which you can see on this electron micrograph. And then also we have the Golgi apparatus, which you can see. So I know a lot of people look at this diagram and they're like, that's really interesting. Like they're a little skeptical sometimes of things in the cell. But these are some pictures of them that have been taken. And yet they're very complex and amazing. It's kind of the point I'm trying to hit home here. And this is a fun fact about this picture. It was made by Dr. McGill, and he's the director of molecular visualization at Harvard Medical School. And he said, although people are struck by the complexity and density of this environment, what we depict is actually very dilute in its concentration relative to a real cell. So this is a dilute form of a real cell. So
you can see the density in those electron micrographs.
Yeah. Well, there's a lot going on in there, and it's all needed for life. All right, now cellular composition. The cells are made up of 4 different types of macromolecules. You have the proteins, which are made of amino acids, the lipids, which are made of fatty acids, carbohydrates, those are just sugars, and then you have the DNA and RNA, which are made of nucleotides or nucleobases. And all types of macromolecules are chiral. So like the proteins, for example, they're left-handed. The lipids are mostly right-handed. Carbohydrates are mostly right-handed, and DNA and RNA are right-handed. So yeah, all the macromolecules are chiral, whether they are left or right-handed. Yep, still chiral. And so this basically means chirality means that they can't be superimposed on one another. So like if you take our hands, for example, you cannot superimpose them on one another. They're fundamentally different. So you can see the little depiction right there. And the purpose for homochirality, it helps the molecule basically have structural integrity, and it will preserve its function. So chirality is very important and necessary for these macromolecules. All right, then the molecular composition of cells. This is just to give you an idea of how big they are and how diverse they are. So they are made of 100 billion atoms. The smallest cell, mycoplasm, that's a bacteria. It's made of 100 billion atoms. One yeast cell has 42 million proteins. The shortest DNA has 580,000 base pairs and 507 genes. And this DNA would be 34 million atoms, just to give you an idea of scale.
That's to make the point that it's not like just by on accident that all these genes and the DNA is necessary. Like these things are necessary for life. Like it's not going to be like replicating and choosing to replicate big long strands of DNA and generating proteins if it wasn't required.
Yeah, it is basically necessary. And then here's some molecules down here that are required for all life, some different elements: oxygen, carbon, hydrogen, etc. All right, so now we're going to get into a little bit of functionality before we go into abiogenesis. So how does it work together? How does it store information? How does it use it? So cellular information system. So this is the central dogma of biology. Basically, information goes from DNA to RNA to proteins. And this is essentially the central dogma. So the DNA will be transcribed into mRNA that will then give the information to be generated into a protein. So then, yep, you basically have that transfer of information going on here that's required. And then the proteins actually make the DNA. So yeah. All right, so Francis Crick was the one that termed central dogma. And the term dogma was actually a misnomer, as it implies an unchangeable belief which contradicts the scientific method. And Crick himself later acknowledged this improper use of the word. So it's not a dogma. Everything is up for testing in science, and we should be able to test and to validate things, and not just say everything is concrete. You should also be using the scientific method. All right, and down here is a picture of E. coli's DNA, and you can kind of see that double helix going on there. All right, and then here's a short video just to show you like the cellular machinery required to reproduce these molecules and help you understand the central dogma. So it's like less than 2 minutes. Let's see if I can speed it up.
The DNA double helix contains two linear sequences of letters A, C, G, and T, which carry coded instructions. Transcription of DNA begins with a bundle of factors assembling at the start of a gene to read off the information that will be needed to make a protein. The blue molecule is unzipping the double helix and copying one of the two strands. The yellow chain sneaking out the top is a closed chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA letter by letter to copy the gene. At this point, the RNA needs to be edited before it can be translated into a protein. This editing process is called splicing, which involves removing the green non-coding regions called introns, leaving only the yellow protein-coding exons. Splicing begins with assembly of factors at the intron-exon borders, which act as beacons to guide small proteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released, and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes remove all introns so that the edited RNA contains only exons, which are the complete instructions for the protein. Again, this is happening in real time. When the RNA copy is complete, it sneaks out into the outer part of the cell. Then all the components of a molecular factory called ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA, 3 letters at a time, and matched 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds, the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message you feed into the RNA.
Cool.
The grid you can see in this microarray.
All right, so you can kind of get an idea of the machinery that's required for cell replication and protein synthesis.
Yeah, that was awesome.
The grid you can see.
Okay. All right, so now we're going to get into origin of life. Dun, dun, dun. Just kidding. All right, so was it a random cosmic coincidence? The scientific community hypothesized that life originated through a random, undirected process known as abiogenesis. And abiogenesis is the idea that life arose from non-life. And here's a fun fact. Panspermia is the concept that life may have originated elsewhere in the universe and reached Earth through space travel. Watson and Crick proposed after discovering the shape of DNA. This begs the question of how it began. So basically, they saw the shape of DNA as a double helix, and they said, wow, this is amazing. And then they said, oh, well, they thought it was designed, essentially. But they couldn't say it was God, so they said it was some extraterrestrials, possibly, that brought this DNA over. So just a fun fact. But if anybody does bring up panspermia, just ask them, where did life come from there? Wherever it came from in the universe, how did it originate there? You're just pushing back the problem. Not to mention in the vacuum of space. Right. All right. So I'm going to go over the history of abiogenesis. So in 1859, Darwin proposed the theory of evolution in his book On the Origin of Species. The term abiogenesis was coined in 1870 by Thomas Huxley to describe the process by which life arose from non-living matter. And then Darwin in 1871 described the warm little pond that life could emerge from. And then we got the Operon and Halide hypothesis in 1929 that said life could have arose from a primordial soup, a mixture of organic compounds in early oceans energized by lightning or UV light. And so right here is just kind of a depiction of some of the things they think could have happened. So you have this abiotic synthesis, synthetic reactions on the Earth, right? Like making these organic compounds, and they have these organic compounds coming from outer space into a prebiotic soup to make these polymers, and then these polymers somehow just made life. And then we have the RNA world, which we're going to get into later, and protein DNA. These are some people think protein and DNA created life. Some people think RNA created life. Some people think amyloids or proteins themselves alone created life. Other people have metabolism first hypothesis. So there's a bunch of different hypotheses, but
either way, if in any of those other hypotheses, life has to completely change in order to start working in a different way, like other than what we see today.
Exactly, right? 100%. Yeah. So we're going to go over some of their theories a little bit later. All right. So I just wanted to give you guys an idea of how much they knew about the cell at this time. So at the time of the proposal of abiogenesis. So in 1868, this is the Fortnightly Review. This was a popular magazine in 19th century England. Okay. And this piece was titled Mr. Darwin's Hypothesis. Okay. So it's basically talking about the cell and the idea of the cell at the time. So I just highlighted this part. It says a cell is a nucleus with surrounding protoplasm. So that's what they said the definition of a cell was. And before this time, they did not have micrographs like at this time. So they were not able to see all the intricacies in the cell. And they just thought basically the cell was just some ball of jelly and a nucleus. So, yeah. All right. And then abiogenesis continued. It was 70 years after abiogenesis was proposed that we can actually see the intricacy inside the cell. So here's some pictures of inside the cell through an electron micrograph. You have the mitochondria over here. You have the cilia and etc. All right. So in 1940, Manfred von Ardeen constructed the electron microscope, which is the reason why we're able to see these things.
Click on
all right.
All
right. So I was looking into the current state of abiogenesis because there's so many conflicting hypotheses that I was trying to see, okay, well, if I go get a biology textbook, I wonder what they're going to say about it, right? So basically the only thing they mentioned was the Miller-Urey experiment, which we're going to get into next. All right. And so this is basically one of the first and most prominent experiments for abiogenesis. It is the most cited evidence for abiogenesis. So we're gonna go into it. All right, so you have Miller and Urey over here on the right, and they attempted to simulate early Earth's conditions. And basically, they were trying to see if they can make organic compounds, right? So they mixed the gases methane, ammonia, and hydrogen in a sealed chamber. And then they added water and heat to create vapor, simulating the oceans. Then they applied electric sparks representing lightning. And then they cooled and condensed the vapor representing rain or condensation. And then they collected and analyzed the tar that was produced. And over here on the right is the what looks like the tar that was produced. So it just kind of looks like a dirty bong, but this is what they analyzed. All right. Okay. So here's the Miller-Urey results, right? This is a paper chromatography, and it's showing these 3 amino acids that they found. So they found, let's see, it's hard to read, aspartic acid, glycine, alpha-alanine, and beta-alanine, but I don't think aspartic acids. So just these 3 right here. All right, and so basically these amino acids, these few amino acids, composed only about 2% of the solid residue that was formed from the experiment. And then the amino acids that were formed were a mix of the left and right-handed forms of those amino acids. So remember earlier how I mentioned chirality and how they all have to be homochiral, facing the same direction? Well, in experiments like this, they get heterochiral amino acids that would not be able to form a protein anyway. Oh, yeah. All right, so this is just the reaction that occurred. It's not that impressive to me. They basically just used methane, ammonia, and hydrogen. They shocked it, and then they made glycine, beta-alanine, and alanine. And these compounds are only like 10 atoms. So you got glycine, 10 atoms, beta-alanine, 13 atoms. But don't forget, the smallest cell is made of 100 billion atoms, just as a reference and comparison. All right, so now here's some criticisms of Miller-Urey. So this one is by Dr. Scott M. Hughes, and he basically said that the concentrations of the components used were basically carefully selected, which would not be like that in prebiotic Earth conditions. It would just be haphazard, molecules. And he said there's no evidence to indicate that Earth's early atmosphere was reducing, and a methane ammonia reducing atmosphere would be fatal. Lightning would have destroyed the organics. He basically said the molecules produced from the experiment would have been detrimental to life forms, that tar that we looked at. And then he said they separated the amino acids from the mix, and then the chirality issue with the amino acids, and then formaldehyde and cyanide are destructive to life, and these are 2 of the things that were produced.
Exactly. So I just want to mention that this reducing atmosphere and the formaldehyde and cyanide are 2 things that you have to do, and if you want to make these nucleotides in the lab, like in a test tube like this. However, you see how 2 different examples of where they would be either, fatal or, destructive to life.
Right, to make these amino acids, absolutely. All right, so some scientists speculate that they did not imitate earlier conditions properly. So here's one quote: "It is possible that the primitive atmosphere was not as reducing as Operin and Urey and Miller had believed." Another one: "The accepted picture of Earth's early atmosphere has changed. It was probably O2 rich with some nitrogen and a less reactive mixture than Miller's, or it might have been composed largely of carbon dioxide, which could greatly deter the development of organic compounds." And then some models suggest that there were significantly lower amounts of NH3 in the oceans at the time. So and also at the time when he was doing this experiment, there were other experiments being conducted that were similar to his, except for they were in an oxidizing environment and they basically couldn't get anything. So if it was oxidizing or even neutral, then you wouldn't have got what he had gotten, which is just a few amino acids.
And like after reading some research articles about what early Earth conditions were most like, it would have found that it would have been highly oxidizing, because if we believe that there was a lot of carbon dioxide in the environment, that eventually makes hydrogen ions, protons, and bicarb, and so it makes the environment much more oxidizing, not favoring the creation of these
organic molecules. 100%. All right, so in their book, in Urey's book, The Planets, Their Origin and Development, it stated the primitive oceans would have been approximately 10% solution of organic compounds. This would provide a very favorable situation for the origin of life. So just to give you a reference, right, the organic matter in our current ocean at the sediment is approximately 1 to 3% organic matter. And they're saying it would have been 10% organic matter on early Earth. So kind of a stretch. All right. Here's a quote from Dr. Albert Bransom. He says, "We claim that living systems we claim that living systems evidence a preoccupation with maintaining chemical specificity, not just having the right molecules to hand, but and especially not having all the wrong ones. Yet in any solution chemistry or any chemistry not managed by a macromolecular machine acting specifically to affect the particular transformation needed, the latter wrong ones invariably overwhelm the former right ones. So remember earlier how I said they only got 2% amino acids out of all that gunk and tar that was produced? So that's basically what he's addressing here. All right, and then now we're going to get into a couple problems, right? The tar problem. So the building blocks assumption is simply dead. This is actually by an original life researcher, John Sutherland, and he's made a lot of prominent papers on original life. So he believes in abiogenesis, but he is conceding this point. The building blocks assumption is simply dead wrong. As a simple thought experiment should suggest if you make up any mix of building blocks products, if you make up any mix of building blocks products you like, however rich, energize it in any way you like, wait as long as you like, nothing we assert having any claim of any sort to being lifelike will ever arise in your flask, not ever. Instead, the chemistry therein will more or less slowly progress towards a Benner's tar. Right, so whenever you have these compounds and you can shock it, you can do whatever you want to it, energize it however you'd like. All we keep getting is this tar, and that's definitely not conducive to life. That's the point. All right, so here's my search for a concise abiogenesis model, which was a lot harder than I originally thought. So I just pulled this from a paper that was published a few months ago. And basically, they were just using a computer code to infer the order of the addition of amino acids. So I was looking at it to see basically where we're at in the original life research. So this was published only 3 months ago. Here's some highlights from it. That the modern genetic code was assembled in stages, hypothesized to begin with early amino acids present on the Earth before the emergence of life, possibly derived by extraterrestrial sources such as asteroids or comets, and ending with a late amino acid. So they are definitely invoking these extraterrestrial sources and comets and meteorites, etc. It's so funny. And then they talk about the hydrothermal vents. They talk about the cyanosulfidic environments that they're having to invoke. They said amino acid abundances obtained from asteroid samples will also soon contribute. So they're waiting on these asteroid samples to see if they find more organics in it. That's so funny. And then they also reference one of Saturn's moons here. And I can't say this word. Can you, darling?
Enclidius.
Enclidius's moon. Yeah. So anyway, and then the hydrothermal vents and then tRNA synthetase, they're kind of wondering how it translated everything into the genetic code as it is. So we'll get into that later. All right, so here's a little summary I made after looking at dozens of papers on abiogenesis on where we're at, okay? So here's the storyline. Get it. Between the edges of a hydrothermal vent, ocean water, and a volcano rock pores form nature's chemical laboratories. Each pore offered a unique microenvironment, some stockpiling amino acids, others stockpiling nucleotides, while a few fostered the assembly of RNA strands. Lipid components delivered by meteorites crashing into the Earth provided raw materials for primitive membranes. Lightning strikes while UV light radiated shallow pools, driving critical reactions. Meteorites crashed near these pools, delivering essential elements and providing energy for certain chemical reactions. Clay surfaces acted as a molecular assembly line, aligning and concentrating molecules in a poured rock near a hydrothermal vent, while magnetic minerals aligned them into just the right configurations. Furthermore, relentless wet and dry cycles in these pools alternately dissolved and concentrated molecules, forcing them to link into more complex forms. Eventually, RNA migrated from the shallow pools to the hydrothermal vents. Tiny lipid bubbles encapsulated the RNA while being able to hold the contents and transfer materials inside and outside of them without breaking down, and began migrating between these environments, scavenging compounds and energy wherever it could. Through this interplay of hydrothermal vents, meteorites, lightning strikes, UV light, and drying pools, the first cell was born. Voila.
Wow, what a story.
Man, crazy. All right, moving on. So basically, I made this chart to try to tell you guys just a few of the conditions and environments that would have needed to be present for the first early organic compounds to arise. Okay, so we got this matramorolite clay that is needed for the formation of RNA. And the environment would be the tidal pools or volcanic ash areas. And then we have the pyrite, which would be needed for the pyrophosphate synthesis. And this would have happened near hydrothermal vents. And that's different than the tidal pools, by the way. And then you have you have this iron sulfur minerals needed for amino acids and redox reactions. This would have had to happen near hydrothermal vents, and then the borate minerals would have formed the sugars, like formose, ribose stabilization, and this would have happened in a evaporative environment. I'm assuming in a pond or whatnot. So anyway, you're just having to see like how they are invoking all these different things for all these different cellular components, but it doesn't really work well together. That's kind of something that we're going to see soon. So here's some of the conditions that I saw when reading these papers, and I just basically did bullet points of them. Okay, so we got these wet-dry cycles. We have the lightning, the UV light, the clay, the comets, the meteorites, the hydrothermal vents, the shallow pools, the porous rock with subsections of environments, by the way, and crystals. So, yeah. All right, so here, this little graph is also showing incompatibilities between the different molecules that would have needed to be formed. So you have the DNA synthesis at a nice 55 to 85 degrees Celsius. And then the protein synthesis would have needed to be greater than 100 degrees, right? So DNA and RNA synthesis would have also needed UV light, but protein synthesis was basically hypothesized to be formed in a hydrothermal vent, not using UV light, just using heat. All right, and as you see, they need much hotter environments than DNA and RNA. And then, let's see. Oh, the proteins require low water activity. The RNA requires wet, dry cycles. And then you got the DNA that needs to be in an aqueous environment. But if the water is like contaminated with too many organics, it's prone to hydrolysis, or basically it'll just disintegrate and fall apart. And let's see if there's anything else. Oh yeah, so the shallow ponds for the DNA and the RNA, then you got the volcanoes for the protein synthesis. And here's some quotes from Original Life researchers actually contradicting each other. So it's saying a requirement for UV light to generate biological building blocks would rule out the deep sea environments or the hydrothermal vents, assuming that they're deep sea hydrothermal vents. But here's another one. To get one's electrons by UV photons at the cost of exposing a tender and unprotected neonatal metabolism to a notorious destructive radiation, UV, we would like to offer a much better deal, hydrothermal vents. So they're contradicting each other. One is saying we need UV light, one of them is saying we need hydrothermal vents. So.
And there's a bunch of incompatibility, and we're gonna get to more of the paradoxes coming up.
Here's the incompatibility paradox. What do you know? All right, so the Maillard reaction. Amino acids can react with sugars like ribose, a component of RNA, in a Maillard type reaction, which could deplete the available ribose for RNA synthesis. So you can't have amino acids and these ribose sugars needed for RNA together, but somehow they would have needed to be together in order for the first life to form. So they must coexist, but they can't be around each other. So get that. It's science, right? All right, so here's a translation paradox in terms of magnesium. So the tRNA folding needs a 15 to 20 millimolar of magnesium, whereas nucleotide synthesis would need less than 2 millimolar. And then you got the RNA bonding that has 15 millimolar total concentration, and it can maintain stability at around 2 millimolar, right? So
I just want to mention how here you said like less than, but it says like great. If it's greater than 2, it'll suppress its incorporation.
So if you have greater than 2, right, millimolar concentration of magnesium, it'll suppress the nucleotide incorporation in DNA. However, in these other cases, you need much more RNA. Much more magnesium, like 15 to 20 millimolar. So this is an inconsistency right here. But basically, the cell will do different things to separate these and incorporate form, and that's one of the amazing examples of the cell. All right, then you got the phosphate paradox. DNA and RNA synthesis. So phosphate has a positive role. It's needed for the activation of nucleotides during DNA and RNA polymerization. However, free phosphate groups, which would have needed to be there for the first cell, such as 3-phosphate on nucleotides, can block DNA synthesis and prevent polymerization activity. And then you have the amino acid and protein synthesis. They have a positive role. They're necessary for the activation of amino acids like amino acyl tRNAs However, they can interfere with the ribosome function, so excess free phosphate will disrupt ionic balances that are critical for ribosome assembly and function. So you need some free phosphate, but if you have it just open in the environment, it will mess up other things, right? So just some incompatibilities. And then here's another fun little phosphate problem. So bioavailable phosphate was required for energy production and as a component of the DNA backbone. However, phosphorus containing minerals on early Earth, such as apatite, calcium phosphate, were highly insoluble. So they're like, okay, well, we need this phosphate, but all the phosphate is bound up in these rocks, right? So that was kind of a huge problem. And then NASA came out and said, oh, wait, we don't need phosphate. We found this bacteria that can incorporate arsenic into its DNA backbone. And then they published that paper. And then basically a lot of scientists kind of squinted at it and was like, what? And so some people tried to reproduce it and ultimately found out that the paper was highly flawed. There was a lot of errors in the experiment and it was just straight up fraudulent. So thanks, NASA. Yep. All right, moving on. So the water paradox. So water is commonly viewed as essential for life. So are biopolymers like RNA and DNA and proteins. However, these biopolymers are corroded by water. For example, the hydrolytic deamination of DNA and RNA nucleobases is rapid and irreversible, as is the base catalyzed cleavage of RNA in water. So this allows us to construct a paradox, right? RNA requires water to function, but RNA cannot emerge in water and does not persist in water without repair. So these are why our cell has these mechanisms in order to maintain homeostasis with everything. So it'll have what it needs, but then not be degraded by it as well. All right, here's an energy paradox. So energy is needed for life. However, raw energy without proper ways to process it will cause destruction. It's like trying to charge your phone using a steam engine or a charcoal grill. It's like you throw your phone in a grill, it's like, yeah, I'm charging my phone. Well, what's going to happen? That cell phone is just going to basically degrade. Or it's like trying to put a Tesla charger in a gasoline intake hole of your car. If you don't have the right energy and you have no way to process it, it can be harmful and destructive. And was there something you want to say about the energy? It's okay if not.
No, just that if anybody has any questions about why some of these reactions are impossible, we can talk about delta G for some of the reactions later.
Mm-hmm. Yep. All right, so here's a UV paradox. So UV light is crucial for the formation of key organic compounds necessary for early life forms. However, it is destructive, right? So excessive UV radiation can actually break down and destroy the very molecules it helps to create. How sad. So yeah, it'll cause what's called a pyrimidine dimer in DNA. So they're invoking it for the energy and the formation of it, but wait, if it's exposed to UV light too long, it totally degrades. So cool story, bro. And the hydrothermal vent paradox. So hydrothermal vents, as you guys saw earlier, are proposed for protein synthesis. However, a study published in Nature Geoscience found that hydrothermal vent conditions dissolved free organic compounds, carbons, into carbon dioxide. They wrote that even the most resilient organic molecules eventually broke down as they circulate through the vent systems. So, yeah. Hydrothermal vents, while they could be necessary, can be very detrimental in some cases. And that's why they have to have these, rocks with pores and wet dry cycles, and it's just a lot they have to invoke. All right, the heat paradox. So heat is a catalyst. It'll accelerate chemical reactions and provide energy for forming complex organic molecules. However, heat is also destructive and can break down delicate biomolecules such as RNA and proteins. So yeah, it can be helpful and harmful. But remember, in early life, you only had one condition it had to form in. They all had to come together and form somehow, right? So this poses a paradox. How does that work? All right, so now we're going to get into some of the cellular paradoxes, the DNA protein paradox. So modern cells require protein to replicate DNA and RNA, but proteins themselves are encoded by these genetic materials. This creates a circular dependency where the components necessary for life to rely on each other to exist, complicating the understanding of how life could begin from simple organic compounds, right? So basically the same thing that makes the DNA is the same thing that is encoded by, right? So DNA makes proteins, gives the instructions for proteins, and proteins actually build the DNA. So which one came first? And so to overcome this, they're trying to say that, oh, well, RNA came first. It's like the mediator, right? Well, if we look here, the RNA, the proteins are also used to produce RNA, right? So, and the RNA gets its instruction from the DNA. So it just kind of goes in a circle and we can never pinpoint. And even if you did have some self-replicating RNA with no machinery to do anything with it with, like it would just be some self-replicating RNA. It's not giving code or instruction for anything, you know? So it wouldn't eventually move to life unless you had other systems already present to do something with it. You need to do something with that information. So, yeah. And that's assuming that all of the RNA molecules, all the nucleotides came together in the right fashion, all in the correct chirality and in the proper conditions. And you're having to invoke a lot to even give them that. All right, so here's Eigen's paradox. So we can't have long strands of DNA unless it can repair itself. And we can't repair itself without long strands of DNA. So this poses a paradox. So here, this was posted in Nature, actually published in Nature. It says he posed the paradox that without error correction enzymes, the length of a nucleic acid would be limited because in larger molecules, mutations would destroy the information content of subsequent generations. But this maximum size or error threshold was too small to encode an error correcting enzyme. Eigen's paradox still challenges theoretical biologists, LOL. And I love how they said theoretical biologists. Yeah. Basically pointing to the origin of life researchers. So anyway, that's funny.
Fairy tale science.
Oh my gosh. Okay, so over here is Eigen, Manfred Eigen, and he's giving a little thumbs up like, hey, try to figure that one out. So yeah. All right, then we have the membrane paradox. So the membrane formation paradox acknowledges that cell membranes can only be synthesized from pre-existing membranes. As an extension, the organelle formation paradox states that the membrane encapsulating organelles can only be produced by pre-existing organelles. And then the protein phospholipid paradox demonstrates that the protein composition in organelles is dictated by the organelle's phospholipid composition. But the phospholipid composition is dictated by the protein composition. So where do we start? You know, where's the starting point? You know, how do we get past this?
You can't.
It's also interesting to have a membrane basically encapsulate this primordial cell, whatever that is. So the membrane would have to basically allow things to come inside and out of it. Because if you just had a little lipid membrane encapsulate it, basically nothing is moving in and out. You're not going to get any more nucleotides to make any more RNA. You're not going to get any more amino acids to make any more proteins. So that's why the cell has transport membrane proteins already in the cell membrane to move things in and out without having to degrade that cell. So, yep. All right, ribozyme paradox. In order for the ribozyme to be functional, it must be stably folded in a complex structure, much like a modern-day enzyme. However, in the absence of a set-aside genome, this same ribozyme would also need to serve as a template for its own replication. Templates cannot have complex stable folding if they are to be accessible to free nucleotides and RNA primers. Clearly, the same RNA cannot meet these two mutually exclusive requirements simultaneously. In the RNA world hypothesis, ribozymes necessarily must have these dual roles. They must have catalytic activity while simultaneously being able to act as a template for replication. Catalytic activity would require stable folding into complex structures, whereas its role as a template requires unstable, relaxed folding. So you can see it over here. The ribozymes, they must have catalytic activity, which requires stable folding. The ribozymes also must carry their own genome. To be replicated, they must have templating activity, and that requires unstable folding. So does it have stable or unstable folding? So the ribozymes, basically, with the RNA world hypothesis, they basically say that these RNA strands, right, double-stranded RNA strands can basically have these catalytic functions, but in order for it to replicate itself, it has to be in a folded manner, right? But in order to get its template, like its information to replicate itself, it has to unfold to give that give the nucleotides, make them accessible basically to come copy. So it needs to be folded to replicate itself, but unfolded to get the information from. So, you know, how does it replicate itself? You know, that's a big problem.
And if someone would say that this thing can fold and unfold like without a problem, they would not be understanding folding correctly. Because like when something folds properly, like it usually takes like other proteins called chaperone proteins to go and like pull on them and get them to like fit right. You know, so folding itself is a big deal.
Right. And so there would have to be 2 simultaneous ribozymes be created, or 1 ribozyme and then 1 functional RNA strand that actually meant something. And then even if you had that, it'd just be continuously replicating this RNA strand, if giving them a lot, right? So they're just replicating this RNA strand, then what? Then what? So how do we get to DNA? How do we get to a protein? Is this encapsulated by membrane? Does that membrane allow for things to transport in and out of it? So even with this is one of their newest findings, this ribozyme, they can self-replicate. That was a huge deal. So, but even with that, there are significant problems. And in my opinion, you're still not getting anywhere close to life. All right, circular synthesis paradox. So basically in the cell, in certain cases, you need something to make something, right? So you need 6 ATPs to make 1 ATP, to make any ATPs, right? You have to have them already there. So it takes NAD plus to make NAD plus. It takes tetrahydrofolate to make tetrahydrofolate. It takes coenzyme A to make coenzyme A. Same thing with FAD. You need it to make it. So here's the citric acid cycle over here. And yeah, for a lot of these compounds, like they already have to be present to be made more of. So poses a huge problem. All right, then the tRNA paradox. The translation system requires a highly evolved set of RNAs, proteins, and enzymes for high fidelity. However, these proteins could not have evolved without an accurate translation system in place, creating a chicken and the egg problem. So basically, let's see, tRNA is on here somewhere. So basically, tRNA is what is the translator for proteins and RNA. So basically, the tRNA will basically match up the nucleotide sequence on the RNA to the amino acid sequence needed for the protein. So it's kind of mediating between these 2 languages. So if you just think about it, if you had some amino acids in a protein, and if you had some nucleotides in RNA, what's going to mediate in between them? Like where is the language going to come from? Like the translation going to come from? It's like sitting in a room staring at a computer code, right, on a piece of paper. Like, do you think that over time, eventually you'll understand what those 1s and 0s are saying? No, you need a translator of some sort. So that's where the tRNA comes in. And that would also have to be there. And it's very difficult. You'll see the tRNA later, how complex it is. All right, so natural selection paradox with Charles Darwin. So abiogenesis will appeal to natural selection to get complex machinery. However, you cannot have natural selection until you have self-replication, which requires complex machinery. So where is this natural selection going to come in at? Chemicals can't naturally select. Chemicals have no goal or function. They're inanimate molecules. They have no end in mind, so natural selection isn't going to help you there. And then if there is some sort of, strand of DNA or whatever, in order to have natural selection, you need this machinery to replicate it and to get feedback. But if that machinery is not there, you cannot get natural selection, right? You need this whole system to be in place to even be able to start choosing, which nucleotides we want or which cells live and which ones don't, in order to continue on the ones that live, etc. So without this machinery there and without the system in place, you can't have natural selection. So that would come in later. Right now, we're just talking about chemical evolution, even though chemicals are totally inanimate and have no end or goal in mind. All right. And then lastly, the time paradox. So complex organic molecules require time to accumulate and become useful through random chemical processes. However, time is the very thing that will degrade organic molecules on an early Earth. So given enough time, anything can happen, just doesn't cut it. Because over time, these precious organic molecules that you just made will totally degrade when exposed to the forces, the elements of,
And you hear this argument so much from people.
Yeah.
I would just
with enough time.
billions and billions of years, it's
Voila, it's there,
It's a fairy tale.
In many cases, it will just totally degrade these molecules they work so hard to produce. So moving on. All right, so this is a funny quote. RNA world hypothesis, the worst theory of the early evolution of life, except for all the others. So this is just funny. There's significant problems with every theory, and that's kind of what I'm trying to hit home here. They can't even come together and decide what came first. Some people say amyloid first. Some people say RNA first. Some people even say metabolism first. So they can't come together because there's problems in each. And so, yeah, it makes it hard to make one cohesive theory. All right, so British origin of life researcher John Sutherland concedes to the lack of progress in explaining a naturalistic origin of life. So it's funny in this diagram he made down here. And if you could see the little purple in the bottom, it says the current state of the field. So we have this inherently favored prebiotic chemistry. Then we have like energy dissipation, major system innovation, optimization of major systems, and then evolutionary innovation up here.
Like all that has to happen before it even gets
Yeah.
above the alive line.
You see the alive line. All this has to happen before we even get to this fully alive line, right? And he says we are here, basically at the bottom. So he concedes it, and this is a prominent origin of life researcher. Please remember that. And I argue that we're actually back here, not even on the chart. Like we're below zero, because actually the more we learn about the cell, the more we see how impossible it is. The more that we learn about all the machinery required for each of these processes and all the conditions each of them would have to be in order to form, the more we learn, the more we realize how could this have ever happened. So that's why I argue that we're way back here. Not to mention the characteristics of life, which I kind of ran through earlier, but living cells need to have a lot of things in order to function. And if you want to say, oh, well, the cell, you could just have like a very simple cell, even a simple cell would still need replication. It would need some form of energy. It would need to translate to carry out protein synthesis. It would need to have a translator. It would need to have somewhere to store this code. It would need to be encapsulated by a membrane that could somehow move things in and out of the cell. And then eventually you got to start making your own nucleotides, because, if we're encapsulated, how are we going to make that work? To get more, you need constantly new materials coming in, and I'm not sure if their membrane would allow for that without degrading. So anyway, the more we're learning about the cell is just pushes back the understanding of how it could have formed by purely naturalistic processes. All right, so this is a statement from Dr. John Sutherland. Over 100 years. Okay, wait, maybe I should read the quote first. But I will just go ahead and read it right now. So over 100 years after abiogenesis was proposed, scientists have yet to create a functional protein from purely inorganic molecules under prebiotic conditions. Scientists have yet to create a fully functioning RNA molecule from purely inorganic molecules under prebiotic conditions. So just understand, like, they've been working very, very hard on this, and they have made some progress in some cases, but in these experiments, they're making them very finely tuned, which actually is evidence for some sort of intelligent design. Because if you constantly need intervention in these experiments, and then they're not just happening on their own, then you're showing the need for some sort of some sort of intelligent design to step in and coordinate things. So anyway, and then fun fact, no living cell has ever been created in the lab from only non-living components. And you'll see on headlines like they created a cell in the lab. No. What they'll do is they'll use an already living cell, right? Mm-hmm. And then they'll change the DNA out or whatever it may be. Cloning kind of happens like that. Basically, you're switching out that DNA of an already living cell. So they're not just creating a cell from scratch. That has not happened. And it would be extremely difficult to make it happen. And I'm sure you would need a lot of intelligent design to make it happen from those scientists.
Yeah, definitely.
So do you want to read this quote real quick?
Yeah, I was trying to read it and find a way to summarize it. But, understanding how life on Earth might have originated is a major goal of Origins of Life chemistry. To proceed from simple livestock molecules and energy sources to a living system requires extensive synthesis and coordinated assembly to occur over numerous steps, which are governed only by environmental factors and inherent chemical reactivity. Demonstrating such a process in the laboratory would show how life can start from inanimate. If the starting materials were irrefutably primordial, and the end result happened to bear an uncanny resemblance to existent biology, for what turned out to be purely chemical reasons, albeit elegantly subtle ones, then it would be a complete recapitulation of the way that natural life originated. We are not yet close to achieving this end, but recent results suggest that we may have nearly finished the first phase. Well,
first phase. But yeah, he is an abiogenesis researcher and he believes in it. But at least he's conceding that they haven't even finished the first phase of it. So, yeah. And it says the prebiotic synthesis of building blocks to which we have devoted so much of our time only corresponds to a small increase in the complexity of the system and to no increase in its aliveness. This is a humbling thought, he said.
Yeah, you only become more and more aware of how complex it is and how more impossible it is rather than getting to some solution. That's it.
Exactly.
So the next segment, we'll talk about a positive case for intelligent design.
Positive case. So now we're going to be giving some positive evidence. So a lot of people try to say, "Oh, well, you just believe in intelligent design because that's just the God of the gaps, right? A God of we don't understand it, so you're just putting God in that gap." No, what I'm doing and what we're doing is taking what we know about things that are intelligently designed and looking at the cell. And we could say, yeah, we see a lot of things in the cell that look like other things that we've designed or that have indicators of an intelligent designer. So it's not a God of the gaps. We're making a claim based on what we're actually observing. All right, so here's some indicators of intelligent design. Complex and specified information. Organized, meaningful complexity. We got the irreducible complexity as well, where all parts are essential for function. Purposeful arrangement, features, designs for specific goals. Fine-tuning, precise calibration of parameters, information-rich features, detailed intelligence, associated information. We got the specified complexity. We got the adaptation of means to ends, where all parts work together for a clear purpose. And we have the creation of new or novel information. Novel functional information suggesting intelligence. So yeah, here's some good indicators of something that is intelligently designed. All right, so we're going to move into a couple cases of irreducible complexity. Do you know if this was the start of your segment?
Yeah, it is.
Okay,
I'll go ahead and take it from here. So irreducible complexity is basically just stating that like one thing can't be formed without the other, that the whole system doesn't work without an essential, component. So the system of like DNA, RNA, and tRNA, making ribosomes, like it all is like super integrated network. So each component's dependent on the other for its function, or else there'll be death or disease. And now I'm going to show you how the ribosome is a complex molecular machine that requires 50 translated, preformed proteins to facilitate the protein synthesis and support the argument for intelligent design. So one thing that the ribosome does is that it translates. And why is it called translation? It's translating between two completely different languages, right? With different spellings and different systems of units. And these have to be like matched up like perfectly for them to work. Mm-hmm. So there's 3 nucleotides in the DNA or RNA, and that corresponds to 1 amino acid. And there's 64 possible codons from the mRNA, and it must be matched up to 20 different, specific amino acids.
Right.
And you can use this little like wheel here whenever when you have like a sequence of mRNA and you're trying to find out which amino acids they're going to make.
Mm-hmm. So you basically have, I just want to make a comment. So you basically have these codons of 3 nucleotides from the RNA that then gets translated to 1 amino acid. So you have to have that translating system already there.
Mm-hmm. And here's like a basic picture of that little translating sequence. This is the ribosome right here, and this is a strand of mRNA. And the picture up here is showing something similar, just like that there could be multiple ribosomes on a single strand of mRNA. And we're going to talk a lot in detail about the tRNA right here, the transfer RNA, which is the one that's actually translating between the codons of the mRNA and then picking out which amino acid we need to use. Okay, so this highly specific translator, it's translating between 2 languages, like I said. And then there's like the amino acid attachment. We'll talk about that more. Okay, so the tRNA, first we should take a look at its structure. It has, so what does it do, right? We already kind of went through this, that it reads the mRNA. It attaches amino acids to one another. And they all carry their own specific amino acid. And,
so you're saying there's like 20 different tRNAs, one for each amino acid?
Yes, this acceptor stem here.
So not only do we have to have one of the translators, we got to have 20 of them because there's 20 amino acids.
Yeah, like we're going to get to it next, the amino acyl transfer charging right here. Perfect. Okay, so like you're saying there's different ones, and that's because of their attachment site. Because of this necessary enzyme right here called amino acyl tRNA synthetase. It's going to recognize this attachment site, and it's going to combine, the one specific attachment site to it. The amino acid it needs. Mm-hmm. So this enzyme, like I said, it combines each amino acid to the necessary tRNA, and if it doesn't work properly, it can recognize that and cut it off and try to replace it with the right one.
Correct.
And I was gonna say, so this is just another example of a necessary preformed protein, and
20 of them.
yeah, 20 different ones, right?
Yeah,
And
it's
then
crazy.
this is a good time to talk about the specific structure of the tRNA itself. There's like a T arm and a D arm, and they have like essential components of each that are necessary for folding and necessary for it to function right. And also you can see here, energy is needed, right? So even if you're imagining some self-replicating tRNA situation, it still has to be in a perfect environment like Taylor's been describing. So the whole ribosome together, right? That's that ball we were talking about, how this whole process is going through. Protein synthesis happens in the ribosome, and ribosomes are molecular machines made of 2 very large RNAs and over 50 preformed proteins. There's 3 different sites in the ribosome. There's an A site, a P site, and an E site. The A site is where the next charged tRNA enters, and then there's the P site where the amino acid from the next amino acids being added onto the chain, basically. The elongation site, it's called. And then the E site is like an exit site where it's kind of resting for to leave and restart the whole process. And I guess I was going to describe here, like this picture up here describes that elongation situation going on. There's a whole like little process going on here, and this is the protein being produced. And as you can see, I mean, this molecule itself is extremely complex, requiring over 50 proteins to function. If any one of those proteins are not there, then disease can occur. The most common one,
And it won't function.
Yeah, it won't function right.
Yeah, but what's the disease?
But there's a disease, Marie Charcot tooth. It's like really well known because of its a failure at this step.
Mm.
And then I just threw this in because I was like, in case you thought, oh, that's it, So this is just pretty much what I've said before.
How many proteins does it have in the RNA? Is it like 30 or something?
The RNA?
The ribosome.
Like the ribosome.
Yeah, the ribosome.
So like over 50. And
Over 50?
That's in a bacterial ribosome.
So a bacterial ribosome has over 50 proteins. So this is needed.
Yeah.
This is needed to create a protein and translate from the RNA to the proteins.
Exactly.
Yeah.
And I mentioned that because they're a little bit different. You can see there's like the small and large segment of the ribosomes are a little bit different from one another. You need these elongation factors for the I think the tRNA to bind to the ribosome.
Yeah, it's a lot.
Yeah. Let's see. Is there anything else? Release factors. I mean, there's just a lot of stuff going on. Okay, so even the minimally functional translation system is inherently complex, requiring ribosomes, tRNAs, AARNAs, that's the charging enzyme, and translation factors. Aminoacyl tRNA synthetases, which attach amino acids to their corresponding tRNAs, are essential for translation. Yet these enzymes themselves require a functional translation system to be synthesized.
Yeah, synthesized. Yeah, so we have they're trying to propose like a solution to this. Like, how could this mediator have came about to mediate between RNA and amino acids and proteins? And basically they were saying, oh, like there could be stereochemistry where like the amino acids like the RNAs and they're on them, and then they start forming this like relationship with one another, and then they could transcribe them. But first of all, when are they still going to have like the translation information between them? Like, that's the first thing. And then the second thing is, remember earlier how I was talking about that Maillard reaction where the synthesis of nucleotides and amino acids, their conditions have to be separate. So the amino acids will react with the sugars like a ribose, which is a component of RNA, which would deplete the available ribose for RNA synthesis.
This is used commonly in medicine to test for fructose in the urine, which is like a biochemistry issue in children.
Wow, that's cool. Yeah. So that couldn't have happened.
Exactly.
And now we're looking at
the irreducibility, right? So we're looking at molecular machines.
That's exactly. Like, we have these amazing machines in the cell that are required for essential purposes. We got the flagellum up here. We have DNA replication down here. Look how amazing that process is. It's amazing.
And it happens so fast. This is like slowed down times 100.
This is extremely slowed down. But yeah, they're absolutely amazing. And then we have the mitochondrial ATP synthase. All right, so we have specific and irreducible complexity. Remember earlier I was saying that was one of the indicators of an intelligent designer. All right, so Charles Darwin stated in The Origin of Species, if it could be demonstrated that any complex organ existed which could not have possibly been formed by numerous successive slight modifications, my theory would absolutely break down. So that's amazing. And I argue that we found many systems like this.
Yeah, it's broken down. Trash. They're in the trash.
Yeah, so a system with several interacting parts, if just one part were to be removed, the system would not function. So that's basically irreducible complexity. And there's many cases of it. The flagellum being one of them. And one of the common rebuttals is, oh, well, there's this other flagellum that we have that required less stuff, but actually, even in that flagellum, it has irreducible complexity as well.
Yeah, I can actually explain. I think what happens is that there's this is like a rotary motor, right? This thing, I think, can go up to like 100,000 RPM. It's reversible, and it functions at nearly 100% energy efficiency. Okay, but what they were talking about was that there's these like kind of like an attack flagellum. I don't know the exact name for it, but it's how like one bacteria or something will go like attack another one. It's just totally different. It's more of like a syringe that is extends outwardly, and it has no rotation purpose.
But I'm sure even that thing has irreducible complexity.
I'm sure it's wild.
Yeah.
And I just want to mention these things up here. Like this is kind of this is in the core of the flagellum, and I think it's super cool. It's something that is kind of related to Taylor's research here.
Still,
There's all these different little parts, like these little arms. These little connector arms are called stators, and there's dozens of proteins that are in this thing, and if any one of them are removed, it doesn't function.
Yeah, it doesn't function. So this is kind of like what the flagellum tail up here at the top would look like if you just cut it in half. So yeah, these are just showing like the microtubules that will radiate through it, and it's an amazing feat. All right, so here's ATP synthase. This was also very similar to man-made motors. So ATP synthase and man-made motors share key similarities. Both utilize rotational mechanisms with a central shaft rotating within a stationary housing, resembling the rotor and stator designed in electric motors. They generate torque to perform work. They are modular in structure and adaptable, such as that ATP synthase can operate in reverse. Additionally, they convert energy with remarkable efficiency, except ATP synthase actually achieves a near 100% efficiency as compared to man-made motors. So these are truly amazing. And here's a little schematic of it up here, just kind of showing how it works. So this is how we generate energy in our cells. Required. All right, so intelligent design and DNA, one of the things is complex specific information. So DNA's organized code acts like digital instructions. And over here is actually a diagram of from inside of the cell, from the mesoderm and ectoderm, basically giving feedback. So they have these like feedback loops already incorporated, and basically feedback on which genes to form more of, which genes to form less of, and the cell, how it resembles computer chips, for example, many parts of the cell.
This is basically telling the cell where it's at in development and helping it decide what cell it's going to become.
Yeah, there's a lot of feedback and regulatory mechanisms.
I think this is like my section.
Yeah, you can go ahead.
And so it's in a similar manner, like we're looking at how there's like informational hierarchy and stuff. So we've talked about how like DNA, makes genes, ribosomes, and the genome. And then there's like molecules that then can form organelles, mitochondria, and cells make tissue, organ systems, organs. Same way your neurons make neural circuits, brain regions, and it makes up your whole central nervous system. But I put these pictures here because I think it's just so cool that, we look at these computer chips and we're like, man, this is super complex. And there could be, a lot of integrations and like switches and a lot of decision making happen. Well, look at this, circuit that you have going on in every single one of your cells. All these things are necessary for you to break down what's like glucose over here, go through glycolysis, the citric acid cycle, and make all these different biomolecules that you run on. And even some of these, well, I just want to say that like these things don't just happen for no reason. Like, and it's really hard to see from your screen, but there's all these little blue words written down on here, and they're all necessary enzymes.
Yeah, so everything is integrated, and we can see that in things that we've created, like the circuit board and computers. So then we look in the cell and we start seeing all this integrated and all these integrated systems, and we're like, wow, that resembles something like what we've made. So, and all of that is necessary. Like they all have to be working in conjunction with one another and giving feedback to one another, et cetera, et cetera. So it's just really impressive.
Yes, indeed.
All right, so here's a paper.
Yeah, this
go
is
ahead.
cool. You talked about like the computational architecture of the cell. The complexity of the cell and its corresponding analogies to contemporary computing systems runs deeper than simply DNA. The cell as a whole has also been viewed as a computational machine similar to multiprocessing clusters with a combination of centralized instruction set and distributed computing elements. The channels of information transfer and transformation in the cell have direct analogies in communication theory and computational algorithms. And
That's
so,
amazing.
like the processing system would be like a gate in circuits, or like the amino acids as basic elements.
Yeah, you got the information code. We have the binary codes in the computers. We got the quaternary code with the 4 base systems in the DNA. You got the primary memory. That would be like the gate in a computer, and the ribonucleotide as a basic element engineered in circuits. So there's so many different examples of just similarities between computer systems and our cell and DNA. Let's see if there's any more cool ones I can find. Is there any more you
Yeah,
want to
no,
share?
I
This is just awesome.
I want to spend more time reading this paper. It's
Yeah.
so cool.
That's awesome, man.
Yeah.
And you got like the basic computing structure. You got the data path in a computer, and then you got the proteins, which carry out the functions. And yeah, it's amazing. All right. So is abiogenesis a viable theory? All right. So theories must have a coherent explanation to whatever phenomena it is trying to explain. And abiogenesis completely lacks this. Okay. There is no coherent explanation to the start of life. And many times I see them appeal to the future, right? Like, oh, well, maybe sometime in the future we'll figure it out, right? But that's actually, fallacious. And ironically, the more we learn about the cell, the more unreasonable abiogenesis becomes. We're actually learning about all this circuitry in cells and informational hierarchy that is needed. So I feel like we're going in reverse. And then personal incredulity, just because you can't understand it, just because you don't want to believe intelligent design or whatever, doesn't make it false, right? Just because you can't understand it, that doesn't make it something that's not viable. So I find a lot of scientists have this where they don't want to say it's intelligently designed, so they'll just go with the contrary. But you should definitely look at all the evidence first. And it is viable intelligent design, or is intelligent design viable? All right, so Occam's razor prefers explanations with fewer assumptions. So intelligent design has one. Okay, and think about all the assumptions they had to make for the first cell that they're still trying to figure out in abiogenesis. Whereas intelligent design postulates one abstract concept, right? So they have one postulate in intelligent design versus hundreds, dozens in abiogenesis. All right, so these prebiotic experiments that they're actually doing are proof of intelligent design because they're having to intervene in these experiments. And so they model the need for information and interaction needed in the origin of life. So whenever they're constantly stopping the reaction, adding new reagents, and controlling the pH levels, they are showing the need for an intelligent designer and something that's intervening in this process. And so, as I mentioned earlier, the intelligent design is not a God of the gaps. It is based on what we do know about where information comes from. And we can observe the attributes of something that is intelligently designed and then see those same attributes in the cell. So we're going off of what we do know about things that are intelligently designed rather than, oh, well, we don't know what happened, therefore it must be God. No, we're not doing that at all. All right, and I'm going to end it with this quote. This is Dr. George Wald, and he actually won a Nobel Prize in 1967 in medicine. And he said, that's the wrong quote. That's the wrong quote. But basically, he was saying that he doesn't want to attribute God to the origin of life research. And so he's willing to believe something that's scientifically impossible over the idea of God. So yeah, that's about it. And intelligent design isn't rejected on scientific standards, but by philosophical ones. And we should definitely not do that. We should definitely hold it to scientific standards. If science is showing us that this looks like it was intelligently designed, that's what we should go with. All right, and thank you guys for listening. I wanted to give acknowledgments. Thank you to Daniel for helping me with the tRNA slides, and to Sam, Andrew, and Peter for helping me with organization. Thank you to Alan and Galatrax for moderating. Thank you to Ether Cause and Earth Awakenings for giving me a platform, and I'm open for any questions, corrections, or rebuttals.
Thank you.
If anybody's still there.
Oh, I think people are putting hands up.
Okay.
think we might have to
Should I
accept
end the stream?
Well, you can just like call them up because we'll keep the slides up or whatever.
Can you join as a listener so I can see who's asking a question?
Oh, sure.
Yeah. Okay, I'll keep the screen. Yeah, I'll just keep it on in case anybody has a question about a slide.
Yeah, excellent presentation, guys. Appreciate the shout out, by the way.
Yeah,
I'm
no
not
problem.
sure if Omnivore wants to come up and ask his question about the flagellum. He said he has a question about. Oh, I'll let him ask it. There he is. Go ahead and bring him up.
So I have heard about the flagellum being irreducibly complex, and I've heard counters to it. You said there's something similar to a flagellum that needs less pieces, but it's also irreducibly complex.
Yeah, so it's actually not a flagellum, but it kind of looks like it in shape, but it doesn't function as a flagellum. So what it is, and I wish I knew the name of it. It basically, it's like an attack syringe essentially for one microorganism, I believe, a bacteria, to then consume another bacterium. And so it's like more like a straw that extends out, and it actually like fires out really quickly. Leading me to believe that it's probably, like Taylor said, there's more evidence of irreducibly complex structures in that thing. But
Yeah.
it is
It was
fundamentally.
What are the walls? Okay, well,
What's that?
what
Yeah,
are
I
the?
didn't decide on that. I just kind of threw that in as a side point where some people say, oh, well, they have this other thing like a flagellum that, has less parts to it. Therefore, irreducible complexity is invalid. But like,
So
necessarily.
okay, well, I mean,
I don't have a slide on that one, but
Bistocles was kind of a jerk to me when I pointed this out, and I want to make it real clear to him that if you're gonna say that I'm just being incredulous about something and then go off on me, Bistocles, you might want to hold your tongue next time. So about about that particular issue, right? That's a particular piece of evidence that we could look at. Well, I mean, you were kind of spicy there, Beastie. So I think it's all fine.
Right.
But you can. So when you're talking about the flagellum itself, I'm making it about you. You're the one who jumped in on that. If you don't want me to make it about you, just stop typing. And I've said all I need to. So are you talking about the macro structures that are created by it, or are you talking about the base proteins?
Just the whole rotary system as a whole.
Yeah.
Like
That's
in a motor. Like think about
If I remove, okay, you can go to the next. You can go to the next one, Gio. I didn't start the drama. If you don't want it, then I
can't hear anybody else except for Omni.
Yeah,
they're typing in the chat. They're typing in the chat to me as we do this.
No, it's okay.
So if you remove proteins from the DNA that creates the flagellum, can you still get a flagellum out of it?
Yeah,
my understanding is you can.
Yeah, I think they've actually tried this before and they found it to not be functional. But that was something I meant to add more about in my slides. I just haven't had time. Like it took me so long to put together this presentation. I couldn't add absolutely everything, but I can hit home on that if I present again on exactly about the flagellum in particular.
Yeah, because there's some I'll put a paper in the chat for all the people who are incredulous.
Yeah, and it's not really just the flagellum. It's many of the molecular machines in the cell. This one's just interesting because it's actually like a rotary motor. And so if you think about a motor and how it functions, if you remove parts from that motor, it's not going to function. So then I guess the question is, how does it
So,
how does it thrive?
but I don't know that. So there's two things, right? There's the DNA code that creates the flagellum,
Mm-hmm.
and then there's the flagellum itself, right? So those are two different things. So are you saying that one of them's irreducibly complex and the other one isn't?
No.
Oh, they're both are most likely.
Well, the flagellum gets the information from the DNA, so they're not like different. Basically, the proteins that make up the flagellum are encoded by the DNA. So you're saying basically, if you knock out a part of that DNA and knock out one of those proteins, will it still work? I mean, yeah, I don't know. And I think that they've done a paper on that and tried to alter the flagellum, and it did not function from my memory. But I don't have a paper on hand to give you.
So
But,
I posted one. I just posted a link to one that I think they were able to do that. So I have to check and make sure that's the right paper. But yeah, I would question that
gene did they knock
example.
out? What gene did they knock out in that paper exactly? Like which protein did they knock out? Shall I ask? Which part of this flagellum did they knock out? I'm just curious.
Yeah,
so
they knocked out COG 1463.
I think that's a species.
Is C
I don't even see
uppercase
that
or
on here.
lowercase?
Uppercase.
COG 1463. And so what did that encode for? Was that the protein?
It's one of them.
What does it do?
Interacting proteins is MOTB. I'm not an expert on this. The analysis, if the answer is I have to have the, a degree in bacterial biology in order to understand whether or not this is a case of irreducible complexity, you might be right. But if that's the case, that's kind of the problem.
Okay, well, if you could just let me know what part it knocked out or whatnot, that's cool. But
and what was the COG part again? COG 1.
COG what?
I put the link in the chat at 30 minutes past the hour.
Okay.
So they have a whole. It's more generally. It's not about knocking out proteins and having the flagellum work or not. That's not what they were doing.
I think they're looking at a different species, right? A different species flagellum. Isn't that correct?
They're looking at flagellas. I don't know. I mean, is there a mobility is achieved by most bacterial species by the flagellar apparatus. It consists of dozens of different proteins with thousands of individual subunits. So it looks like they're just talking about flagellums in general. Is there one particular flagellum you're thinking of?
Like, are you asking us like which flagellum are we referring to?
If you're referring, well, she said they're talking about a different species of flagellum.
I think that you're addressing the same thing that I addressed, which is that they said there's like a different type of flagellum altogether, but that type would still be, have irreducible complexity. And the purpose of irreducible complexity is like, okay, so if we don't have like, how would these start forming this rotary motor if it didn't have an end in mind? Like there's no goal in mind. So why would it continue to move towards that goal if it had no function? So if you look at this system right here, the flagellum.
I'll tell you right here, what they modified was this thing over here on the side. The stator. I knew it. I knew that if you knock something out, I looked it up. With the gene you were talking about is the stator. Okay,
well, that's just one. That's one of several.
Yeah,
I mean, they did a whole bunch.
It's like saying like what there's probably
we took the door off a car and
exactly.
It still worked.
Right. Like how?
Okay, so it's not the motor; it's the whole car.
Yeah, like even if you take the transmission off the motor, it's still going to run. You know what I'm saying?
Right.
And so,
wait a minute. Originally, we were on the motor, right? We were just saying it's pieces of a motor, not it's pieces of the whole car. So, what piece can you take off of a?
We're talking about the essential rotor in the center, like the rotary motor, and the whole thing as a whole. Because how would any of this start to form if it has no end in mind? That's the real point here. Like, how would it even start to build if it couldn't function?
So, I've heard that argument too. I don't know.
That's what it is. That's the argument. Let me give you the point of this, right? The point is that there's no purpose for it to continue to evolve something that has no function. And without all these pieces, even if you want to knock out, the door of a car, without all the pieces of the motor of the car and essential components, it's still not going to go. You know what I mean?
I don't think you can go.
There's no. There's no reason for it to continue to evolve something that has no function without all the pieces there.
So is it
You know what I mean?
is it possible that a partial flagella confers some advantage?
Well, think about it. Like, is there a partial motor, right, that's missing crucial elements? Can it still
Well,
function? That's the question.
Yeah, but we went from a motor to a car with missing a door. So I
No,
don't know.
Because this
the
side
thing you're
piece,
talking about is this purple bar over here. It still would be able to push like energy through it and stuff and still form. You're talking about something that is associated with it that might help it be more stable in the membrane.
Yeah, but
I mean,
they
go ahead and knock out one of these components of it and make it and watch it work. You understand what I'm saying?
okay. All right. Yeah, but they did a bunch. That was just the first of
then
several.
you should have the then tell me which ones they knocked out.
The MOB, the thing on the very most
On the side,
outside.
yeah. But it's not even really about that. The whole
FLJ, FLGE2, FLIE. I mean, all of these show
criteria that we're referencing with the different
this paper is not about knocking out proteins. That's not what it's about.
I thought you
It's
just
about
said
it's about whether or not the flagellum can function with modifications to those or removal of those proteins. If a protein doesn't function, what happens to the flagella? So they didn't. It's not attempting to disprove your.
Did they find it was
thing.
functional? What was the results of the study?
Yeah.
The result was that it was still functional. Yeah, from what I understand.
Okay. Well,
from the MO.
Hey, can you give me the name of that
Mo-
paper really quick? I'll look up. I'll go.
Protein network of bacterial motility.
I sent
Wait.
you both the paper in Discord DMs. You both have it in your DMs.
Yeah, they'll
on
send it
Discord.
to us.
Oh, thank you so much. Yeah, I'll
I'll
check
look
it
that
out,
one.
and hopefully I can give you a response before the live stream ends.
Okay.
Okay.
It was
Galatrax.
Thanks, Beast. That was good.
I'm glad you got your question out.
Oh, I did.
We got Robert V. Utah up here. Did you want to ask a question as well?
Yeah.
Yeah, so I want to ask a question. The name of the space is called Scientifically Dismantling the Myth of what? Making vehicles? What myth are we?
Abiogenesis.
Biogenesis?
Abiogenesis, yes.
Abiogenesis, I see.
Were you here for the presentation?
Yeah, presentation and contribution. Yes, sir.
Yes, indeed. So we're just talking about how life is super complex and it's awesome, and it was designed by an incredibly intelligent God because he made all these super cool components that we get to appreciate. You want to?
I don't think intelligence really sums up what God really is capable of. It's beyond that,
Yes,
so
indeed.
right. Because God is the Alpha and the Omega. So He all
Amen.
things He has created. And what I take from that as a Christian is that we can't limit God. Many Christians try to limit God. For example, this big discussion about the shape of the earth. Many Christians are limiting God's power, saying that He created a little flat plane for us to just live on with a dome over it.
Robert, we're trying to keep the presentation to stuff regarding the subject of abiogenesis and their arguments surrounding it. So if you have any questions about that, go for it, brother.
Yeah. No. I was just responding to the gentleman's question there. So how do you guys see, understand abiogenesis? Like,
so what we're doing?
how
So
do you guys see
abiogenesis,
that?
like I think the word abiogenesis means life from non-life, and so we've been arguing, really just absolutely demolishing abiogenesis by showing that the positive case for intelligent design. Like, why should we be very reassured that life shows evidence that it was intelligently designed? And so we sort of systematically went through like a bunch of different things. But if you have a question about any of those things, we'd love to talk about them.
The thing is that remember, God created us from the dust, man from the dust, right? Women were created from a little bit more than that, right?
Mhm.
So what's dust? What's the dirt? Dust is just, you know, non or living matter. So, you know, so the abiogenesis does not, you know, eliminate intelligent design. I think that's the confusion, right? So we have to be careful as Christians what we're communicating to people and so on.
Okay.
So God knows He's the creator.
So,
You understand me?
you know, I kind of like am picking up what you're putting down, right? And it's sort of the idea that maybe you're saying that, you know, God grabs the dust and that breathing life into it makes life. But like there's many people who don't want to talk about God and are terrified of him, frankly. So they employ all their possibilities to come up with some scientific reason as to try a scientific reason to ignore God. But what we're more saying that, we know for a fact that it couldn't have happened without God. Does that make sense? Like,
Okay, knowing. Okay, so Christians are, scientists are going to always push back on that. But the deal, what we have to understand as Christians is that, you know, God's timetable and God's time frame, because the scripture even talks about it. You know, we're on His timetable. So, the 7 day creation is not 7 of our days. You know, it's a process that God is going through. And when they talk about evolution again does not eliminate God. It doesn't, right? So we have to understand that the creator process and timetable is different from ours. I know it's a controversial thing that I'm saying, but we have to not limit God. We have to not do that.
Then don't say he can't do it in a day or 7 days or 6 days. He did it in 6 days. So we're not going to limit him because he did incredible things in each day and in 6 days in total, right? And so we're definitely not trying to limit him. And another thing is that you're right. I believe that if someone was to somehow come up with some evidence that evolution is true, that also does not eliminate God. Because I believe that God intervenes and gives us every breath and is always doing things. In fact, I'll tell you something. I'm so glad we're on this slide. I read a whole paper about how this ATP synthase right here, it doesn't make any sense. Like the energetics of putting like an ADP with one phosphate group, combining 2 other phosphate groups to it to make an ATP, an adenine triphosphate, doesn't energetically make any sense. Okay, so this process is going on. We know that ATP has to be made. However, it's as if it wants to function in reverse. But what it's doing is it actually is creating energy. And so this obviously challenges like a big portion of the biochemistry textbook. But I'm arguing that like God is driving our ATP synthesis like every second. He's your breath of life.
No, absolutely. Come on, he wouldn't do that. You really thinking that God is in all of us turning our wheels? No, come on, no.
Oh yes, 100%.
No, absolutely
100%.
not.
Yeah.
Absolutely not. Because if he was doing that, then we do not have free will. He
Oh no,
specifically,
he's giving you the ability to have free will.
independent us.
Listen, I'm not telling you he's making controlling everything you do and you have no free will. What I'm saying, he's giving you the opportunity to have free will. He gave you life. He gave you a mind. He gave you the ability to have free will. I'm not,
Yeah, but he's not. There's no way he's in our mechanics, biology doing it. No, he already did that.
I'll use your words. You said not to limit God. Thank you, sir.
You. But that's a limitation.
Don't limit God.
Okay, sounds good.
Okay, so I was just reading this paper that Galatrack sent me from Omni, and basically they were able to find 164 genes that they knocked out that completely inhibited the function of the flagellum. So if anything, this gives more credence to intelligent design because they showed multiple examples of how if you knock out one protein, it does not function.
So they gave an example of knocking out 160 different ones,
and it stopped working.
and it stops working. So
that's exactly my point.
The level of irreducible complexity really just got larger. You see what I'm saying? You see how you dig and you dig and you dig. Oh, we're looking for answers. The only answer you're going to find is that, irreducible complexity is seen in all sorts of things, and it becomes even harder and harder to fight for abiogenesis.
I got a question real quick. Could you go back to the pages with the paradoxes? Because there are so many of them, and I just think that's like, those are so amazing to look at.
I know, I love this. I'll just start from
Asking for a friend.
I guess. So we have the time, natural selection, the tRNA, circular synthesis, ribozyme, membrane, Elgin.
Oh, yo. Sorry, I cut that dude off in the middle of his thing. I thought I was up here to unmute to announce what was happening or whatever, but I was saying, if you guys don't want to continue like a meta conversation about, the limitations of God, but like this is the actual physical mechanisms that are put forward as the explanation, and you guys are dismantling them to a degree to where it's not even a tenable position to say like, oh, but if we like invoke unlimited time, maybe God, no, God didn't make it retarded, right? Like this is a very clear set of instructions that have to, like, it would have to be spontaneously done or not. It's not an overtime issue. We're not limiting God. But anyway, like we're looking for specific, refutations to the mechanisms that are put forward rather than like appeal to infinite possibility through God. So anyway, you guys are welcome to, anyone's welcome to come up and ask questions and stuff, but
Any
please
comments,
don't be
questions?
endlessly circular.
Yep.
Yeah.
Good point, Alan. Honest. Just what's your guys' most, like, in all the paradoxes, which one do you guys think is like the strongest one against abiogenesis out of all of those you presented?
It would be easier to say if they actually had a coherent theory.
Yeah,
It's just the incoherence of their different theories and the situations that would have to happen for each.
The most common one is the time. I hope I didn't cut you off.
paradox,
But you'll hear so many people,
right?
even our grandparents are like, oh, well, you never know what happens over billions of years. I'm like, yeah, a bunch of things degrade. You ought to know that.
Yeah.
I don't know. She asked, what's our favorite one? I mean, I don't know. There's so many. I guess I'll take back to like.
I like the information, like the tRNAs, like how even if we hypothetically had all these amino acids there and we hypothetically had all these nucleotides that were in the right chirality, suitable for life, and even if we had everything, like what's going to translate between them? And like they're working so hard just for these organic molecules. And like even like if you did get one, you have to have all these systems in place to do something with it. That's the point. Like everything has to be working in coherence and already there for it to be functional. And they're like really trying extremely hard just to make like a functional RNA just from inorganic compounds. And like assuming that they even did, like then what? You have to have all these other things to make it work and do something, So that's one of the biggest points that I have.
Gotcha. We do have RG the artist here. He did want to ask a question. RG, you got the floor.
Hey, thank you very much for that wonderful speech. That was nice. I learned a lot tonight. So Taylor, I was wondering, with how our our environments are always feeding us light information, and I'm wondering through your studies about how that maybe the continuing feed from our outside environment, how does that affect and influence the cells in DNA?
Wait, I'm sorry. Like, can you repeat your question? Like, so how does the outside environment affect our cells?
Yeah, like the light, like through the skin, and then through the rest of the processes, I guess, like from the moonlight or like the magnetism or different spectrums from the sun, I guess, UV and stuff like that.
How does it influence life?
Yeah, like the continuing information from your natural environment. Yeah, I guess is that
Oh, the sun and the light is actually very important. Push full screen. Sorry, I'm trying to get this right quote up that I was trying to end with.
Was it the George Wald one? Yeah, do that one.
Yeah. It'd be nice if we can make it a little bigger. But just to answer your question, I was just gonna say sunlight is very important and key for like things like what's it called? Vitamin D. And then remember that researcher that you were telling me about that actually says like the light that you eat in is more important than what you eat?
Jack Cruz.
Jack Cruz, he's
Yeah.
awesome. Yeah, Shane sent me a video from him recently.
Awesome.
And yeah, it's amazing. Like, and also light, like biophotons. That's like a kind of a new area of study for biology. Like we're seeing that we have these biophotons that could be sending information within the neurons, because the information, how quick it gets sent within our neurons is like incredibly quick. And some people say that the chemical theories, right, even though they are playing a role, is not fast enough to translate and transmit the information that we need, you know, and whenever we're functioning.
Mm-hmm.
So basically, there's theories about biophotons that are being conducted through like the microtubules, which is what I study, to basically transmit information in the brain.
Yeah, another thing about that, and this is quite a while since I read this paper, but I'll just try to keep it short. They basically took slices of different species brains from mice to like maybe dogs to like monkeys, and we're shining light through the same sized thickness of the slices of brains. And as you were basically like moving up in species, like these more intelligent species were absorbing more of the light, so the light was more redshifted. And what they're saying is they simultaneously were looking at the glutamate receptor. So glutamate receptor is stimulated on a neuron and makes it depolarize and will then continue an action potential to another neuron. Well, the point is that light will activate the glutamate receptors. Okay, so one neuron can carry light down it like a fiber optic cable and then shine on the next neuron in the same way that it action potentials.
And it's like transmitting information.
Yeah.
I've heard about that. That's really interesting. So, you know, things like that just show how much more amazing the cell is than we know.
Yeah, yeah.
And here
I would encourage. I was going to say I would encourage you guys to look into also bioplasma. There's a lot of good studies going on with that as well. Yeah,
that's awesome.
What is that? Bioplasma?
I'm not sure.
Well, like your auric field extends out from your internal bioplasma, which basically is the building blocks for your neurons and neural pathways. Bioplasma. Check it out.
That's interesting. Thank you for sharing. I just wanted to read this quote from earlier. So this is George Wald, and he actually won a Nobel Prize, and he studied at Harvard. And he said, there are only two possible explanations as to how life arose: spontaneous generation arising from evolution, or a supernatural creative act of God. There is no other possibility. Spontaneous generation was scientifically disproven 120 years ago by Louis Pasteur and others, but that just leaves us with only one other possibility: that life came as a supernatural act of creation by God. But I cannot accept that philosophy because I do not want to believe in God. Therefore, I choose to believe that which is I know is scientifically impossible: spontaneous generation leading to evolution. George
At least he's honest enough to admit that there's a little bit of a philosophical bias there.
We have to give him credit for that. But this is what we're dealing with here. I feel like a lot of people just have cognitive dissonance when it comes to intelligent design. Even if everything is pointing to intelligent design, it's just glaring in your face. They're like, no, it couldn't have been. It has to be another way. Can't believe in that, you know?
Well, yeah, what are the implications if there is a God?
Exactly.
Jeez.
Exactly.
And if anyone else has any questions, just raise your hand and we can bring you up and you can ask any questions. How long do you guys think you're going for? How long would you guys like to go for?
We can go however long you guys want.
Yeah, I mean, as long as y'all have questions and want to talk about it, or just discussions, comments, you know, like
any feedback,
anything we could have done better or whatever.
And Dan here is in medical school, so he could probably answer a lot of health questions if you have any.
Have some random questions we can talk about together.
Right now, you are currently streaming Discord. I have a question. When did you guys first start questioning abiogenesis. What was the moment for each of you?
When we stopped streaming our computer.
Like,
So
when did you first start questioning abiogenesis?
So I first hold on. Can we stop the stream?
Yeah.
I don't even know how to do that. Oh, you just pushed that one. Okay. Cool. So I first started considering abiogenesis kind of like it's always kind of been in the back of my mind, like how life got created. And then as I mentioned, I asked my 5th grade teacher how life arose, and then she said, "Oh, well, chemicals must create more chemicals." So I was like, that was ridiculous. I mean, the way she was answering, I knew that she didn't know what she was talking about, and she was kind of thinking about it herself, like, that is an interesting question. But yeah, I've always thought about it, and it's just been kind of common sense to me that something like that could not just happen by accident.
Hmm.
What about you?
I'm really trying to think about it, and been an evolution, like my understanding of abiogenesis and my personal feelings about it probably really started like in biology 2 class, but man, it really like was solidified in biochemistry because like I showed that biochemistry map, and I remember like learning about like in biology 2, learn about a bunch of different body parts and stuff and like how the body functions. And I was like totally amazed by it, thinking like, how is this all like how could all this be an accident or anything? But then in like biochemistry, you learn that these chemical reactions, they have like delta G constants or whatever, where the reaction doesn't occur on its own unless you have this enzyme, which is a preformed protein. So then you learn that the enzyme is this protein that is only made in the cell through this complicated process that we talked about.
Yeah.
And so I said, well, shoot, if the thing needs itself to function, then it can't come about on its own.
Yeah, like you need an energy input for a chemical reaction to happen, but these chemical reactions do not naturally happen. So that's why we have enzymes that come and lower that activation energy for the reaction to happen. But without the enzymes, nothing's going to happen. That's why they're having to invoke these crazy circumstances and prebiotic earth and conditions in order to make these reactions happen. They don't happen naturally. And if they do, they usually just, organic molecules just degrade over time and don't form anything. Like in nature, if we're just looking out into nature, like aside from any life, like considering prebiotic earth, everything moves towards non-life. Everything will degrade and have entropy naturally. You're having to jump over so many hurdles in order to make this theory work, as you can see. It's not something that happens naturally.
And I'm guessing the answers you guys got during your education was not satisfactory, so you decided to look a little deeper, and then you came up with this awesome presentation. To which, did you guys have any more comments on the paper that Omnivore suggested?
Well, Omni suggested that paper. Yeah, I went and looked at it, and basically, they found 164 genes that whenever they knocked out, the flagellum did not work.
Yeah, like I commented on that slide and I said there was like a dozen or a couple dozen proteins that if you knock out, it doesn't function. Well, it turns out it's 160 genes instead.
Even more.
So if you knock out any one of those 164 genes, it ceases to function.
Yeah.
Ah, gotcha.
Okay, so a little bit of self-debunking then. Did you guys know that abiogenesis comes from the prefix a, not bios, life, and genesis, earth, origin, or creation? So not life origin, not life birth, not life creation.
Yeah, and that actually contradicts like with the laws of biology. Like, there was a funny scenario that happened when I was in school. Basically, we had like a biology quiz, and one of the questions had to do with basically the laws of biology, how life only comes from living cells, right? Life only comes from life. And then I put, no, that's wrong because abiogenesis. And then I was incorrect. And then I was like, but wait, didn't y'all say life came from non-life on early Earth? And then she was like, yeah, I guess, like, and gave me the point back. But I think maybe she wasn't a fan of it either.
Oh, you think she didn't entertain abiogenesis, but she may have had to for the curriculum?
I know from my experience, a lot of my biology professors, like kind of under the table, didn't support abiogenesis. And some of them openly saying that, God created all this. No way.
Yeah.
So
it's like it's not the best idea, but it's what we've got for now. it's a work in progress idea. And then, I had some comments on your presentation too. I think, first I want to say thank you guys for presenting. You two formed a watertight premise. The conclusion follows. It tracks, it's sound. You guys have proven that abiogenesis as an explanation for the origin is untenable, and it's not a sound argument. It doesn't track. It's not sound. And then, as far as the positive claim section goes, which is the second section, usually the question is, what would it take to falsify your premises? But your entire presentation is covering that, so it seems like there'd be too much. There'd have to be way too many things to falsify it. The odds are stacked against the abiogenesis. So the so-called scientific theory of abiogenesis sounds more like special pleading, appeal to possibility, a la philosophical bias.
Exactly. Yeah.
Takes a lot of faith.
They're moving in reverse. And it would, one of the things that would make me question if abiogenesis was tenable is if they were able to actually put organic compounds together and get life. But or even get like some moving towards life. But what we're seeing, like I said, is they're like they are a lot of times they're incompatible. And you have to have special enzymes moving these reactions and compartments in the cell to separate these environments they need to be in. But without that, you just have them all mixed together, and that is detrimental. But that's what would have been needed. So that's why they have to make all these special cases of this had to form here and this had to form here, and then how do you get that all to come together is the question.
I really like the graphic that you guys showed, which had that mall arrow that points to it. At the mall it says "You are here," and it points to that slice at the bottom.
Yeah.
We did have a hand, but it went away.
I see.
I do have one more question, if that's okay.
Oh yeah, for sure.
So it's kind of a broad question, and it's for both of you that did the speech here. So as creationists, what are y'all's future plans in this field that you're, talking to us about, and what would be the area of most impact that y'all would like to work on next together? To make an impact and to make a statement, maybe, as creationists.
I was actually talking to Dr. Brian Miller the other day because he was looking through my PowerPoint, and they do have creationist researchers, and I would love to participate in that, but it doesn't really affect researchers like currently, right? Because like most of the time you're just in the lab working on your own research, following the scientific method, and anything that's like following the scientific method and like showing that something's real is like tenable, right? And I don't have a problem with that. But it's the crazy claims that are like have these just such impossibilities and that are so far-fetched that they're like positing as fact to people that I have the problem with, because we should be utilizing the scientific method to, find out what's true and what's not.
Yeah, that's true.
Yeah, absolutely.
I guess I'm currently training to become a physician, and hopefully will one day become a general surgeon. And I think that understanding that we're all intelligently designed and we're all, brothers and sisters, helps you to show compassion and love for each other in a way that you wouldn't do otherwise. And so just I hope that it helps me like become more loving and impactful to others in a positive way.
And a lot of physicians are actually like that. They believe in intelligent design. There's a lot of scientists who also do. They just get kind of pushed to the side because like there's a overarching, materialism that's hovering over science, and we should definitely try to remove that and just look at everything empirically and say, what is the evidence actually pointing to, even if it's not what we want to conclude. So we should be more open-minded in that fashion.
Awesome. So stay optimistic, and thank y'all for doing this again. One more question, last question, and I'm gonna fall back and land the plane here. So I don't know if y'all have done much research in like the forces of like the weak forces or the strong force, like in magnetism. I'm sure that plays a role, like as far as holding these structures together, which are super tiny, right? But super intricate as well. So, do y'all think about that much, or have any opinions on that? Like the role it plays?
I'm not sure if I can really comment on the role that it plays, but I know one thing as I was thinking about the like different like reactions that happen between like functional groups and stuff, and I mean, I guess I just don't know how it works in this conversation, you know, but there's like certainly so many different conditions have to be present for like all the different necessities of life to come about, you know.
Yeah.
And different, you know, I don't know, that's it.
Yeah, so I was just gonna say like the magnetic field, like birds and stuff like that can pick up on it, and like they do like migration and stuff, and they hypothesize that there's these like magnetic proteins in their brain that allows them to, based on the magnetic field, be able to know which direction to go. In their eyes?
In their eyes, yeah.
That's
So maybe they
interesting.
can like, you know, see a
That's
compass.
weird. That's weird. Or see like some type of magnetic field lines, or not lines, but just like something. I don't know how it functions. I don't think scientists necessarily know completely either, but yeah, magnetism, very interesting. So I know in terms of birds, they can utilize magnetism, but I don't know about humans. I'm sure there's some magnetic field that permeates around us in some way. And also like the quantum properties of microtubules, they have like frequencies. And they found that if you alter these frequencies or expose them to different frequencies, they can actually either stabilize or degrade. So like you could be prone to that in your environment.
All right, we have Fundy up. Floor is yours if you have a question, comment, etc.
Hey, thanks, man. Yeah, great stuff, guys. I don't know. I got here a little late, I think, but did anyone ask you the age-old question, what came first, the chicken or the egg yet?
Yeah, we covered that one.
Yeah.
I got
maybe the RNA, the what's between a chicken and an egg.
I always
The
answer.
chiclet.
I've always answered this question with the chicken because you know nothing. Any animal's always going to lay the egg of itself, right? So if you're talking about a chicken egg, the chicken has to lay a chicken egg, and so like
what did God create first?
Exactly.
Probably the chicken.
You know.
Yeah.
I hope that answers the question. You need to have
absolutely.
You know.
Right. Just poof, fully formed chicken. Of course, every chicken came from an egg, and every egg came from a chicken. So it's yeah, quite the conundrum. I got a more serious question though. Do you guys see anything in your research that points towards life actually having some beginning, some amount of years ago? Or is it on the table that life, like living beings, is something that could have always has been and always will be, and didn't necessarily have a first created thing, but basically just always living things have always been around?
Where, on Earth or in the universe?
Wherever. Just I guess the universe.
Well, in some whatever whatever your definition of life is, then yeah, there's something conscious that's always been here. I believe in an immovable mover, right?
Mhm.
There's something that had to move, that had to create, and that was never created. Because if you continue to ask, oh, well, who created the creator? That's like the infinite regression paradox, and you never have a cause to the effect. And so I think that there's always been some sort of life per se, and some sort of intelligence, and something that designed this place. But in terms of biological life, I'm not sure how long that's been around. I don't know if we were the first humans ever created or not. Biblically, yes, we would be, but just depends.
Cool, thank you. Last question. I don't know much about it. Just wondering if you've ever heard of I forget the guy's name, but he's famous for this concept of the morphogenetic field. If that's of any interest to you guys.
I'm gonna have to look into that.
Do you know anything about it? Like
morphogenetic.
Just the basics. You guys would probably recognize the name of the guy who like coined the term, but it's kind of the idea that either in the DNA or something that there's basically like an electromagnetic field that is then filled in with the flesh, that like there's sort of like a predestined shape that guides like the cells to sort of fill out like coloring within the lines of a children's book or something, and that the lines are sort of I'm not sure I can put it much better than that. I'm not too much of an expert myself on it. Just wondering if you guys had heard about it, but yeah, check it out. Maybe
it seems like it might be kind of related to like energetic healing or something. Like you have like energy fields like in your body, and it drives development and stuff. Like in the development of the embryo. Well, we didn't even really talk about that, but man, that's amazing. You really want to get, shaken up. Go like look at how we develop in the womb. It's incredible.
Someone posted in chat another paper called Magnetic Bones in Human Sinuses. So I sent that to both you two. You can check it out.
That's dope. Thank you for sharing. Cool. All right. Well, I've really enjoyed being here. Does anybody else have a question before we head out?
A quick one. Yeah. How do you guys know you wouldn't be happier believing in random cosmic coincidence?
Well, I mean, would you be happier thinking that you came from a monkey? Because I wouldn't. I can't stand the whole monkey man theory. It's ridiculous.
You say?
I'd be lost. You see, people having a purpose is essential for is very important for being fulfilled. And it's we should all be glad to know that there is strong evidence that you were designed and built for a purpose, and you were designed by an incredibly intelligent God who loves you. I think that should only benefit everyone, right? I don't understand why you would be opposed to that.
Because people just want to act however they want.
Yeah, probably.
Thank you guys. Presentation was awesome. I love watching you guys. You have great synergy. I like how you guys go back and forth. It's awesome. This is all really great. Thank you guys.
Yeah, thanks for having me. And if anybody has any questions, you could just DM them to me and we'll get back with you. Thanks for being here.
Thank you.
All right, so I think we can wrap it up there. Gio, if you want, we could move everyone down to the live stream channel now.
Husband for Valentine's Day.
Yeah.
Yep.
Wait, is Galatrax in here?
Yeah, are those orchids?
Yeah, they're orchids. The whole orchid plant.
That's amazing.
Yo, orchids are like one of the most unique flowers out there. I love them so much. Good choice.
I told both of you guys to get the other flowers for Valentine's Day, and then I explicitly said, "But don't tell them I told you."
I went and picked some outside. That's the best I could do.
That was funny, Gally.
I went and picked some flowers.
Yeah, she
Came from the heart.
I know, it was awesome. Like I like got to her and she like presented her little flowers that she got outside, and it was like so cool. Like I really loved it.
That's amazing.
Love Gally's trying to be a little hitch. I love it.
Yeah.
And I was like,
go get her flowers. But don't tell him I told you. And he said the same thing to Dan.
I was in the lab working so hard, and he just came through and he brought me some good food, and I was just like, "Aw."
Oh, that's
like
better
the
than
first thing I
flowers.
got was a Bible. Like I got her like another Good News version Bible, which is like her favorite because it has the Apocrypha in it, and it had like a cool cover. It has like a I don't know.
And the name written in the back was Tayler. So it belonged to Tayler.
Oh,
and it was so
cool.
cute. And it's like a blue and it's cute. Anyway. Well, I wasn't able to read the live chat like during the presentation, but if there was any like unique comments or any rebuttals, like I was kind of hoping to get more rebuttals.
I tried. I was fishing. I didn't really get many.
Where are they at?
I mean, it's hard to rebuttal like 7 paradoxes.
That's so true.
There was one rebuttal I could read it to you. We asked him to come up, and he didn't want to, but I could.
He was like, "Questions are pointless at this point." I'm like, "Okay, bro."
Why? Why did he say that?
Yeah, so
Uh,
his position was these things don't just happen for no reason. I guess he was quoting you guys. He said, "Yes, certainly not in the time frame you're capable of observing. I'll call God of the gaps. You'll call time of the gaps, which means you're denying that time has existed way, way longer than you did. And now there's a bunch of analogies like, can partial motor work? Sorry, analogies are not science. There are literal tons of scientific papers published on this topic, and the only way to argue them is to deny all and everything that does not align with religion, because there's simply this need to believe in religion, and people can't resist fulfilling this wish to their own minds. So yeah, truth can sometimes be too painful to face. I guess I can only envy believers because they don't have to deal with reality." That's your rebuttal.
Can you
You know-
message that to me, please?
Yeah, please do. Like,
And I want to go and I just want to
Oh.
respond to a couple of those things because that's I mean that's a typical
it's so
feedback.
funny. One thing I was thinking about was like, it's easier to be fooled than it is to realize that you are fooled. You know,
Hallelujah.
And they will sit there, and I can just say, like, I wish he would just read his own message and say, well, isn't that what I'm thinking about with origin of life science? Like, it's just a filler where like we're always going to be constantly looking because it must have happened somehow. But then you just find out a bunch of reasons why it couldn't have happened.
Yeah. Okay. So he says things just don't happen for no reason. Yeah, certainly not in the time frame you're capable of observing. Well, the thing I kind of mentioned the time thing. They couldn't have had infinite time in their primordial Earth, because organic molecules degrade, know, and so like that posits the same question. And they should be able to observe these reactions happening because they would have had to happen in that time frame in the early Earth. Because if they were exposed to elements too long, they would have degraded. So they should be able to replicate some of these things in our time scale.
Yeah, so also I said, all right, so if you have a question, why don't you ask it? Or if you have a rebuttal to what was presented, request a mic to be heard. He said, because it's a bit pointless to be asked nonsense questions like, who gave you
What?
life? And then trying to explain that you cannot just personify the origin of life and make such assumptions while asking a question. I don't even know what that means. But that's what he said.
It means
I got
I'm a
another.
coward and I don't want to hold my position. That's what it means.
I got another question for you guys. I don't know if you view it this way, but you just you use the word like degrade, and maybe I'm putting words in your mouth, but if like genetics and DNA and stuff like that kind of get corrupted by the environment and stuff over time, do you think that means that like Adam would have like the purest like genetics, and that since then things kind of only get like more diseased and corrupted?
Yes, indeed. So
100%.
Taylor and I got into this after our study of like Genesis, which could be like a big presentation on its own. I think we were talking about it earlier, maybe. But so you can trace back the Y chromosome to like your paternal grandfather, right? Your dad's dad, etc.
You can
All the
trace
way
the
back to
one
one man. And you inherit your mitochondrial DNA, which is in your mitochondria. We could do a presentation on that, but it makes the energy. But in there, it has separate DNA itself, and you get it from your mom. So you can trace back your mitochondrial DNA from maternal grandmother, and they can trace back the maternal, mitochondrial DNA all the way back to one woman. And so
With the Y chromosome too.
yeah. So and also they both trace back to one person each. But then on top of that, the Y chromosome is degrading generation over generation.
Yeah.
And so you're right. Was Adam the optimal man? I mean, I believe so, right? If he was made by God, then he would have been optimal. I mean,
But then also, like it said, death will, if basically you will surely die. God told him if they partake in the knowledge of good and evil, and that's a whole presentation in and of itself again. But just for the assumption that they just ate a fruit, just for now, like they came into mortal bodies, and I believe that they now had the ability to die in the flesh. Like I think that they were going to probably be immortal before that event happened.
Genesis
Whereas
3, right?
Wasn't our first estate the light body, right? Isn't that scripture?
Well,
I just don't think that they would die beforehand.
Well,
I wonder
Genesis
what-
6:3 says, and the Lord said, my spirit shall not always strive with man, and for that he is also flesh, yet his days shall be 120 years.
Mm-hmm.
Mm-hmm.
Right.
Well, what you guys just explained is actually an answer to one of my previous questions that maybe got a little misunderstood, or maybe I didn't say it right. But when I asked, in your research, does any of it point you guys towards the conclusion that some amount of years ago there actually was a first, living entity? And so I guess your answer would be yes, that the mitochondria and tracing back lineage stuff, you said all back to one Y and one X, or one male and one female. I guess that does mean the answer would be yes to that question.
Yeah. I think that I don't know. I just
Guessing
think
how
that
long ago it was is a different question, of course. How long ago was that thing? That's probably impossible to tell.
Wait, somebody did the
6,000
research.
years? Do you guys like that idea?
Did the research and traced it back to the biblical times. Like,
Yeah,
I haven't looked at that study yet, but I have heard that somebody's traced back how long have humans been around, and basically it matched up with the biblical timeline.
like somewhere roughly around 6,000 BC,
I can't remember.
like 4 to 6,000. I don't know.
I don't remember, but yeah, it was basically like no more than like 10 years.
I've got a documentary you might like. It's on the nature of plasma that I kind of brought up earlier. I'll drop it in the live stream text and in y'all's good time when you want to learn a little bit about it. There's a 3-part series, and it's eye opening for sure. Yeah.
So
There's a lot more things we can. Yeah, go ahead.
I wanted to finish responding though to this dude, Nevi. And he says, I'll call God of the gaps. But it's funny because I already addressed God of the gaps. Like, we're not saying it's God because of gaps. We're saying it's God because there's indicators of an intelligent designer. So we're making a positive claim, right? Like that we see intelligent design in the cells. We're not just saying, oh, we don't understand it, therefore God. So if anybody says God of the gaps, just please know how to give them a rebuttal. And then also, I don't know what this part means. Way longer than you did. Now there's a bunch of analogies like, can a partial motor work? I mean, that's what it is. And you saw in that paper that Omni brought up. Can it work? Look, they knocked out 164 genes and found that the flagellum was either paralyzed or not functional or severed or whatever. Like, so basically they found all these necessary proteins that are required. And the point of it was that, I think that he misunderstood what my actual point was, which is like, why would it continue to evolve if it doesn't have a function? Why would they continue down that route to build this quote flagellum that they knew was gonna be a flagellum, which the first life would not know? It's just kind of adapting, right? And like, how would it keep forming that without an end in mind, right? That was something we mentioned. There's no end in mind, right? But like, so they wouldn't continue down that route. But we see it, and there's been arguments basically saying that the cell looks like it has top-down design, not bottom-up design. With all these circuits that have to be in place, it indicates a top-down, not bottom-up design.
Yeah.
Isn't it ironic though that like they have to basically pay attention at every step of their process in order to keep their experiment going, which is like ironic. Like, isn't that a fine-tuning problem?
Yes.
That is evidence in and of itself for intelligence.
That's so funny.
That's what I mentioned. Their own research and how much intervention. They acknowledge how much intervention. Go read the materials and methods section to these papers. I'm telling you, it is so long. Like, I tried to add some, but I'm like, okay, this is just too much. Like,
Too much.
But this is the next point that he was saying. There are literally tons of scientific papers published on this topic, and the only way to argue them is to deny everything. What have I denied? I went and read those papers. I went and tried to find the best papers for Origin of Life to say, yeah, abiogenesis is real. So I tried to find their best evidence, and I looked at their papers, and what I saw was basically them trying to go above and beyond to form these small steps. Like, for example, like making a nucleotide, right? They're trying extremely hard because it is very difficult to build these nucleotides, especially in the right chirality. But so basically they'll go to extensive measures just to build out one step. And they are intervening throughout the process. And it's just a lot of fine-tuning in the experimental design. And I have read their papers. If he has any paper that disproves anything I said, please bring them to me. And I just posited, and I think he was responding to this posit I made, like, we have not made functional proteins or RNA purely from organic compounds from scratch. Now, what they will do is they'll take organic compounds like we saw in the Miller-Urey experiment and form a few amino acids, or, just assuming all 20 amino acids. So yeah, they're making that, and then but they're not using that same experimental condition to form a protein, right? So then for the next step, they'll say, okay, we took these amino acids and putting them on this clay and doing these like thermal like heating and cooling and wet and dry phases. They got these amino acids to bind to one another,
Isn't
right?
it only 13 atoms compared to like the simplest cell being like 100
yeah,
million atoms? So it's not
100
even
billion, yeah.
yeah, so it's not even analogous. And you're
It
saying
isn't,
that's
but
their best evidence, right? A
yeah,
dirty
they just
bong.
do stepwise. Yes. That
Oh,
is their
Dirty
whole
bong.
experiment. But it's stepwise. They're doing everything in stepwise fashion. They're not doing a one pot shot
Yeah.
where they're putting everything in a pot and getting like these functional, different types of molecules.
Exactly. They're building off of one another. They'll cite one another's sources, but they're jumping over like a cliff.
They're not starting from scratch.
You know, they're jumping over like
Or
a
like,
barrier.
or like one of the most recent and profound findings was this self-replicating ribosome. They said, oh, we get ribosomes, RNA to self-replicate. Okay, well, you had to use our RNA polymerase from an already living cell and tweak it to make it start doing this RNA replication. And then you're going to say self-replicating ribosome. So the thing is, like, you got to understand, they're putting an already living ribosome. I'm sorry, they're putting an already living polymerase in there. Like, and then on top of that polymerase, they have other RNAs for it to replicate, and it's not like, so they've actually retracted the term now, self-replicating. And then also they have to break apart other RNAs in order to do this. They found that it was breaking apart a bunch of RNAs. So, and they'll make a claim, like a profound claim or title, but then once you go read the methods and materials and what they use, you'll realize it's not what they say it is.
When you guys say it's like a step-by-step process, are you familiar with the Lockheed Martin F-22 Raptor? It's a plane.
Yeah.
Yeah, would you say that it's more likely that was assembled, like that becoming assembled, via random cosmic coincidence, like a tornado flying through a scrapyard that has all the parts?
all the time. Yeah, of
What's
course.
more likely? Is it
I'm
that,
looking.
or would you? You're hot, Mike RG. Would you say that's more likely, or would you say that human beings, the design of human beings, is more likely?
Man,
there's so much
that's a
more
good one.
complexity. There's so much more complexity in a cell than this, than a
the
jet
raptor.
engine. What do you think?
Wasn't that oh, sorry, go ahead, Fundy.
That was just an awesome answer to the whole. Oh, you find a watch in the forest. You think the forest put that watch together, or you think someone made it and dropped it on the ground? Yeah, but your answer to that was hilarious. I love it.
So for the self-replicator, right? It says the odds of choosing 250 correct proteins for the minimum self-replicator equals 1 part in 5 times 10 to the power of 961 to the power
Given
of
enough time.
250. That is 10 to the power of 240,424.
Wow.
That's the odds or whatever for a
I know.
self-replicator.
If I do another presentation, I would probably include those numbers because I think that helps.
It's insane.
But people are gonna say, oh, there's not one chance. So I don't even want to give them that one chance.
Yeah, don't even.
Because I don't even think that one chance is actually possible in purely with purely inorganic matter and in primordial Earth, like in the conditions that it is. So, like, we haven't even got there to a functional protein, like.
Yeah, according to
You
their
know, it's
own.
really.
Oh, sorry. Go ahead.
You know, it's really funny about the give it enough time argument is that they haven't given themselves infinite time in the argument because they gave us a date of the beginning of the universe, and they also gave us a date, more importantly, of the beginning of Earth. So all these things, in this one and yada yada Googled 0 exponential power chance, right? That'll have to happen within the past like 10 billion years, or because it couldn't happen in the first whatever billion years because Earth is a molten lava rock or something, right? So it has to cool down, the water has to come, and only then can you start like doing this whole process. So the whole give it enough time argument is invalid because they've put themselves under a time constraint.
Well,
yeah, time is not on their side. That's the general rule of thumb.
Time degrades the molecules that are required to basically need time to basically make themselves or whatever. So it's that's the paradox, the time paradox.
Like I said too, with natural selection, you don't have natural selection unless you have like the self-replication system already in place. So you can't say, oh, well, that replication system got there by natural selection, because there's, you need the replicators in order to make the DNA, which would then undergo natural selection. So it's like, circular, right? Unless you have the mechanisms to self-replicate, you can't have natural selection. And you need natural selection to get the mechanisms that self-replicate. So
Yeah, I mean, even in their own paradigm, what you said, Fundy, it wouldn't even work either. Like, even in their own paradigm, they say that the chemical assembly power, like the amount of chemical reactions in the universe is like 10 to the power of 120, right? They would still have to account for 240,000 more. Like, odd. Like, it's so ridiculous. Like, there's not even
And
enough
it did it.
material universe for her to do
And
it.
blind, unmotivated matter accidentally created life, right? Accidentally. That's not even the right word for it, but you know what I mean?
yeah, and
lightning
the
too.
reason. Go ahead, Beast.
No, my bad. Go ahead, bro.
No, I was just gonna say, like, the reason why I really like this topic is because I feel like it's the best argument for a designer. Like, it's like unrefutable. Like, it's like, the best evidence for a designer. I mean, yeah, you could say the Earth, the creation of the universe, but the fact that we can see indicators of intelligent design here and compare them with our own things that we've designed, it's just amazing. So I really want people to like have this knowledge to be able to refute these atheists that say, we just could have came about by accident.
True that. True that. Hey, I did drop those docs for you, Taylor and Dan. And
Oh, yeah.
Dan, this one was on the fourth state of life matter, the bioplasma. I think you inquired about that. So it's
Yeah.
very eye
I'll
opening.
tag him for you, bro. There you go.
Thank you for showing me that, because it's interesting. It makes me think about something I heard like from Jack Cruz, and he talks about how like important it is for us to get sunlight. And like, so sunlight contains light, which is like almost optimized at activating these it's called a chromophore in the mitochondria. And so, I mean, I don't know if I could like publicly speak on this or like really represent this, but like basically from my understanding, I think that means that we are getting energized essentially from sunlight. And if that's true, and then also you combine that with what people talk about like with grounding, and I guess I haven't really looked at the science of that either, then like what if energy like flows through us where we sort of
Yeah.
absorbing energy from the sun, and we're connecting to the ground, and eating whole
Yes.
foods, man, that's real life there.
Yeah, they showed this doctor and his patient. She had problems with her circulation, like her red blood cells,
Uh-huh.
and so they did an exercise where he would show her cellular cells and everything, like her structure, and it was showing how the blood cells were moving very slow. But then when they went outside and grounded for 10 minutes and came back in and did the same, recordings, oh man, uh, her energy was like amplified and her blood was flowing so much faster. And it was just a simple and repeatable scientific method to show, this connection you have with the earth. Yeah. Yeah. So it comes through you and it does amplify everything for sure.
And it's so funny. This guy was saying that the truth is too painful to face, like referring to me and Dan. That's hilarious because why didn't you come up here and ask questions? Why didn't you bring these points up? Like, it seems like the truth is not on your side, right? And so it seems like maybe it's too painful for you to face, and you're just projecting. And that's why you refuse to just look at the evidence for what it is. You don't even have to extrapolate. Just look at the evidence for what it is, and if life could form from inorganic compounds, and look at all the research that they've done to try to make it happen, and tell me who's denying reality.
Are you
Evolution is-
addressing me or?
No.
If you're the one that made that comment, then yeah.
No, you're a Christian, right, Robert? No. Talking
No,
about
not Robert.
Yeah.
Who was that? Just said that.
Even Nevin, they're not even in here. I wouldn't even bother.
Yeah, I just wanted to finish responding, but yeah, that's about it.
Is there anyone that's actually a proponent of abiogenesis in here, or an atheist who wants to stand behind evolutionary proponents? Anybody? Like, she's looking for a rebuttal. Like, she wants to know if there's anything that she misspoke about, etc. Some even
She
negative
wants that
feedback is
smoke.
still feedback.
What,
You mean that smoke?
I'm the kind of person that says, evolution doesn't disprove God. I'm that kind of person. You understand? It's hard to not see that in our environment, biologically, how things change, how things grow. So if it's too hot, right? Or check this
Hey,
out.
Robert, just
Hold
quick
on. Let me
question.
finish. Let me
Quick,
finish. Hold
quick
on. Hold on.
question, Robert.
Just let me get it
Do
out.
you
Doesn't take
acknowledge the
long.
difference between evolution and abiogenesis? Are you
So,
acknowledging the difference between these two things? You're conflating it by the sounds of it, mate.
oh yeah, no. I know the difference, right? I know, but evolution's precursor is abiogenesis, right? But check this out. If there is a plant in your house, right? You can watch how the leaves or the tree or the plant leans to where sunlight is coming from, right? You see that the plant needs sunlight to make energy, right? So if sunlight is hard to come by, the plant is going to do whatever it takes to find the sunlight. So evolution, it may change, it may
Well,
evolve
that's
to
just the
be
plant
true, to
pointing out.
find out and seek out how to get sunlight.
Wait, like
Right? These things don't disprove God. We have to get out of that whole mode. You know what I mean?
No one's saying that, bro. No.
I think it's just a semantics thing. Like, what is the meaning of evolution? Like, scientists are proposing that one organism is becoming another organism, as in, a chimpanzee becomes a Homo sapien or something like that. As an example. But like
No,
an
that's not
organism.
what they're saying. That they're not saying that at all.
Hold on, let him
Okay,
finish,
well,
bro. Let him finish.
you can correct me. I mean, what you can explain what you think evolution is.
Wait, hold
I
on.
mean,
I just want to mention about the plant. Don't you think the plants were just designed like that so they can get sunlight? Like,
yeah, they spring up.
if God designed the plants to want to survive, wouldn't they want to face towards the sun?
Yeah, but what if there's an upheaval in the environment, like an earthquake causes the plant to fall below ground?
Then
Instead
it
of the
springs
plant,
up.
the plant might not die, but the plant might over a millennia develop the mechanism to get back to the surface to find sunlight. You guys,
Can
you
I
understand what I'm saying?
It's just gonna die,
can I ask
dude.
can I
If a
ask
plant that needs
quick
sunlight
question?
falls into a dark hole, it just dies because it doesn't have what it needs.
Or
Robert, the issue is that a plant is a plant.
it may adapt to its new environment without sunlight. You guys understand?
You're talking about
Okay.
natural
Then it
selection, right?
already had that ability.
One sec. I mean, Robert, do you agree that a rock did not evolve into a plant? Just quickly.
A rock? No. What do you mean by rock? Are you saying
Well, there's a
rock?
rock. I'm shorthanding. Essentially, an arrangement of minerals and molecules have sort of found themselves in a specific arrangement in order to start self-replicating. This is sort of considered to be the drive towards life, as you refer to with the plant reaching for the sunlight, essentially. So then, I mean, you're trying to point out sort of, what would you call that? Sort of like a basic life, what is it trying to do? It's got a compulsion, a meaning, a purpose. It's trying to achieve something. But a rock can't do that, and neither can all of these minerals and molecules do anything akin to what a plant can do. And yet this is what the proposal of abiogenesis is that somehow these molecules
That's
have
not
found
what.
themselves in a
Absolutely not.
self-replicating system.
No,
I mean,
abiogenesis is not saying that. No, abiogenesis is talking about matter. Matter is not rocks, right? Only matter is
minerals.
anything. It could be biological material. Matter. You understand?
No.
Did it
No.
spontaneously combust into life, Robert? or was it created by like a designer? It would seem like just the presentation we've watched, you saw at least some of it. It seems to have like properties that require it to sort of come at least largely in like fully formed. These are not sort of what's the terminology, like random sort of chaos order that emerges into sort of a perfect design, essentially.
Yeah, they're not starting out simple, they're starting out complex.
Hence
Right.
irreducible complexity.
Right. It's the material, right? Because remember, like for example, two hydrogen molecule atoms come together, right, to form a hydrogen molecule. That molecule under immense pressure will turn it into helium, right? This process causes an immense reaction, a large explosion. Lot of byproducts come from that might
Yes,
cause
fusion.
other atoms to behave differently, right? So what I'm saying to you is God could be the spark of all of this process. You guys thinking that God went in and literally put your hand on and put your fingers and your fingernails? No, he sparked the process. He's the catalyst. He's the energy that started it all, and then it carried on its own. Now, me as a Christian, I do believe.
Hold on. Let someone respond to
Yeah,
that, please.
yeah. There's got to be a response. Unless someone else wants to go, I got a response or a question.
I mean, I'll
I
summarize.
think we should let
You're
Dan respond.
Y'all can go
Okay.
ahead though, because I have something to say, but I want to. Y'all can go ahead.
Okay, I'll make it real quick. That still sounds like, I mean, Tarek put it simply as a rock. We all knew what he meant, but all that's happened is you've clarified that you're saying that, well, don't call it a rock. It's a combination of hot solids, liquids, and gases and plasmas. But still, that was Tarek's point. You believe a combination of hot, liquid, solids, plasma, gases found a way to randomly be arranged coincidentally to just start doing the whole life thing.
Yeah.
It's almost like what is it? God created the environment for which this complexity
Exactly.
to merge together, but had no
Exactly.
personal hand in actually forming the molecular structures necessary. Which is, I mean, you're an abiogenesis believer then at that point. So we can let's go. Let's get him.
The precursor.
Dan, you want to go?
The precursor is God. He's the one that caused all these things to go into motion.
So I
Once
guess
again, you just okay. Go
you
ahead, Dan.
just said like you just had to bring God in for the system. So I guess you're not an abiogenesis, like it's something. I mean, Taylor and I went to a conference about intelligent design together, and there was some time like there was differences of opinion of like who God is or how does he work amongst us. So it wasn't necessarily a conversation about God, but all about how there had to be a God for us to be how we are, and for all the different animals, not just humans, but so we can address evolution too. But what our presentation was on today was how the start of life cannot happen on its own. It can't come about by inanimate objects. And even the system, okay, let me tell you the system that you're referring to. You're saying, oh, God just cranked up the motor of this car and it was running. Well, it doesn't necessarily work quite like that because of something that is called orphan genes. An orphan, right? Someone who doesn't have parents. Okay. And there are so many orphan genes. I wish I knew more details about this, but we're talking about thousands of genes across like every different, like all these different species and different, complexity of life. Okay. So much so that like we could not have come from another like another kind of monkey like animal or something, because we have thousands of orphan genes that make the human unique in itself, that it
Oh,
can't
that's
evolve from
interesting.
another. They don't like, and these genes they do stuff, they function. Even things in DNA, like we can break down junk DNA too. I can't stand that term, but you hear it all the time. Those things are there for one purpose or another, even if it's just scaffolding, even if it's just there to fold in a certain way so that another necessary gene is in the right position to be reproduced, transcribed, and in order to become mRNA. And so these orphan genes are super cool. I think we would include it in a future presentation if I talked about this again. If we were addressing evolution, that's an evolution topic. So even the system that you're referring to has to be, I'm telling you, it has to be designed. And I think it's just makes a lot more sense to me that God just designed everything as he wanted it, as it should be.
Dan Taylor, I disagree with you guys on a few of like your views or whatever, but I do admit I think I'd end up sounding a lot like that last quote that Taylor read if we actually did engage in it, where I'd end up saying like, yeah, you make good points, but on my own gut feeling, I just have to reject the apparent facts you're presenting and go with what I am philosophically biased to believe in. So yeah, I'd be afraid to like go toe to toe, even though I'll actually choose not to do that so I can go on believing the way I would like to pretend it is.
Yeah, at least you'll admit it. You recognize it in yourself, but you can't keep the blinders on forever. Like one day, and I know I can't show no science with this except personal hand anecdotal accounts. But one day we'll be outside of this body where you're blinded by the physical, and you'll see
Hell yeah.
everything as it is true. And we'll stand before in judgment, and we'll understand beyond what we're capable of now, and you'll have no
I hope so.
philosophical argument to hide behind.
Yeah,
Um,
I really hope so, man. I'm not sure if that's true, but I really would like to believe that after death, most of us will get all the answers to the questions we never found the answer to while alive, and that more complete understanding will be bestowed upon us after death.
Look, this life that we can observe in nature with so many new things that I've been finding out since I decided to donate almost all of my time to research in just different topics, right? And what I've seen and what these new sciences are going to start showing, especially in the study of plasma, is that first of all, I don't know who all has heard of Kirlian photography, but what it does is
Yeah.
it photographs your energy output, right? Your aura. And so that's plasma for one. For two, we're told we have a spirit, that the spirit was breathed into us. It was also called life, right? So a lot of people in the like just different studies anyway, there's something that they call the light body, right? That's supposedly part of us. And I think also that it's contained within this closed system called our physical body. Now, the reason for me rambling all that was to say this, that there's something called bilocation that people with telekinesis or telekinetic kind of abilities, and you can be in two places at once. What in the quantum world they're calling entanglement. Well, people that can astral travel or whatever, which I don't fully believe in, but there are studies that these people are being in two places at once and describing what they're seeing. If that isn't a spirit or a soul, I don't know what is. But I'm excited for the future of those kind of revelations, which would prove a creator hands down, which I already believe in a creator, but I think that would be extra cool. But anyway, I'll start
Can
back.
I warn you? Can I warn you about that Kirlian photography a little bit, man?
I'm not sure what you mean by warm, but sure. Yeah.
Oh, well, if you don't know what I mean, then you might be falling for it. So There's like the Russian guy whoever to develop like Chilean photography in the first place. But then there's all these people who are calling themselves Chilean photographer, set up at your local flea market as a little stand or at a mall or their own self-run shop or whatever. And what it started out as is not what people are doing out in the world in the more popular sense of what's being called that. And if anyone ever asks you to put both your hands on a metal sheet or on a tabletop, and then they're going to take a photo of you and show you an aura around your body, that's not authentic original Chilean photography. That's just like basically using the temperature hands through like a software on a computer to create sort of a mood ring effect, and then that is just like transplanted in a sort of like an automatic Photoshop to put a colorful sort of arrangement of colors around a photograph that's taken of you when that has nothing to do with the real stuff. And basically that's just the same. It's basically just a glorified mood ring photography technique. The whole aura photography thing when it comes to Killian, that's all a hoax. But Killian photography that is a real thing in its original authentic sense though. So that's the warning. Don't get the things people are doing with what it actually originally was.
I got you. Yeah, thank you for that, man. But no, I've never had or been around people that are like out in public like that. But no, thank you for letting me know.
But the thing is, as humans, we give off thermal energy, right? And that can be photographed, right? If you look at us through infrared, imaging systems and stuff like that. So, try not to look beyond that. There's a lot. There's even like the human heart has a toroidal field. These things can be imaged and detected, right? So try
Yeah,
not
I
to
got you.
think
I got
that
you.
those things are like the spirit of some sort, right?
Yeah. As well, speaking of toroidal, the spins of had well, let's just say small structures have a field around them, like the charge, and so yeah, the toroidal is from the micro to the macro for sure.
Right. Like the MRI, which is actually a small form of spectroscopy. These are very powerful imaging tools to really, take different views of the biologics, right?
Guys, I just want to say I'm about to crash out. It's 1 o'clock in the morning, so it was a pleasure listening. Great job, Tayler and Dan. I hope to see more work from y'all in the future. So, and everybody have a good night. God bless.
Thank you.
Yes, ma'am.
Yeah, thank you so much.
Yeah, Dan Taylor.
Thank
I
you.
would really like to believe that physical reality and like fleshly beings are something that always has been and always will be. I understand you responded to that idea earlier with the whole like turtles all infinite regress sort of idea response in a sort of debunking fashion. And so that's why I was asking you that other question is like, does anything point towards a certain amount of years or the fact that like there was one thing? And I wouldn't be able to argue against because I haven't done the research about what you're saying about like tracking the X and Y stuff back to like a single source. So that sounds like really good arguments that I wouldn't be able to argue back against. So it definitely is, like I was saying, reminiscent of that last slide Taylor read. Like I just like would probably have to reject that and just go on believing what I want to believe more in that sense. You know what I mean?
Yeah, thank you, Fundy. I had a question for Dan.
Yeah.
Thank you. Name is John. Thanks for your presentation, and thank you, Taylor. Thank you for all you do. I just had a simple question. Do you find you get much equal pushback from Christians, Muslims, atheists, or agnostics when they are presented with your work?
Not necessarily.
No, not really. I mean, I'm open to pushback because I mean, not to say that I have a lot more to learn about all this stuff, but I haven't really felt all that much pushback, to be honest.
That's exciting.
Yeah.
Keep up the
Yeah.
great work. If I have
Yeah.
any other questions, I'll shoot
We
them back.
want more pushback, more from the scientific perspective, from other, religions or whatever. I mean, I think that the light shines in the darkness, and the darkness shall never overcome it. And that if you keep learning and you keep digging, that the truth will reveal itself.
Oh yeah, that leads to my next question. I did forget initially. How far away would you guys estimate abiogenesis being created by individuals in a lab? How much more
It's going
time do
in
you
the
think they
road.
have, if it's even possible?
Impossible.
Yeah, I definitely am not going to say when it's going to happen because I think it's impossible. I think that the reverse is happening. That rather, like the more we learn, sure we can find out, oh, this thing here worked. But in the process, you're going to find more things that say, well, this has to be constructed in an intelligent way and stuff.
And
Yeah,
incompatibilities.
finding incompatibilities and like revealing more paradoxes.
There's a middle ground. I'm not sure if you're familiar with this concept, but what are your opinions on the capability of someone creating a homunculus?
Homunculus?
You would have to
Like a small man in the brain?
like when you we both are thinking of homunculus like as your motor and sensory homunculi, which represents like the motor cortex that it controls
Right.
different
That's a
parts of your
borrowed
body.
term. Yeah, no, I'm familiar with like the psychology homunculus or anatomy homunculus, like you're saying, different lobes of the brain being associated, and that it's like a model of like conversation, basically. But what I'm actually talking about is like the alchemical homunculus, which can be pretty simply defined as the artificially or the man-made man, not through like sex between man and a woman, but the artificially man-made life form. And but the thing is, the alchemical homunculus can use like a man's sperm. It's just sort of a one-sided process. You're not allowed, because obviously there's like test tube babies or whatever that we might even now be able to grow in, I don't know, some sort of artificial womb. But the homunculus is like the alchemical homunculus is like the asexual, I guess you could put it, man-made man, the artificially created life form.
Okay, so we brought up a couple different things. I'll ask you: is a chimera something that you might be kind of referring to, like the mixture of man and animal, or
No, that's really similar. That's like another alchemical creation, but no, more of it. No, just a human, just like growing a human in a lab. But the thing is, you're allowed to use sperm. Is the idea?
Well, you could do that now. I mean,
a
that's
cell,
IVF. That's IVF plus an artificial womb. That's already a
but no
thing.
egg. But no egg.
No egg. No. A sperm is not. It doesn't. The sperm is destroyed in the egg. All it delivers is it's actually. I have to go review this. It's really complex the way the genes interact with one another at fertilization. Yeah.
Well, what do you think about those
Let me just say
snakes
real
that
quick.
apparently?
Well, let me just say real quick. The homunculus is usually depicted as like a little man. Yeah, it's like a little guy that like runs around. It's like a miniature human made by another human. Like in certain ways, and like kind of bringing life
I'm
out of the
sorry,
alchemical
I
process.
can't get the like sensory homunculus with like giant lips and hands out of my mind.
I know that's the
The
first version
homunculus,
of it you've heard of.
there's actually homunculus paradox, which is also known as the infinite regression paradox, where basically it was like, okay, well, if basically it's like, how are we conscious? Oh, we're conscious because this little man inside of our head is running around doing everything. And then it's like, oh, well, who's inside of his head? And then it just keeps going on infinitely. So it never actually solves the
Right.
problem. So that's like, yeah, the homunculus paradox. But I don't know about a chemical homunculus. I'm not sure.
Right.
I
Well,
just posted
what do you, have you ever heard?
I
You
just
ever heard of
posted
those
the
female snakes that reproduce without ever mating with a male snake?
Yeah, that can happen.
Some animals also store sperm for a long, extended period of time, so they
Ooh.
appear to have never
Amoebas.
reproduced that year, but they may have been fertilized. They were holding sperm from 2 years ago. You know.
Yeah, but that could happen. I mean, they have
Okay.
things that can self-reproduce. I mean, that doesn't
Yeah.
disprove anything, you know.
The what's it called?
I think
Yeah,
the
they have
Komodo
amoebas.
dragon is kind of similar. And sharks. I think sharks, some species can do that, but they're just using their DNA, you know.
Or there's some worms that can do this, where you cut them in half and then they
Regrow.
they regrow.
Mhm.
That's
Yeah.
different. Trying to think of the name for that thing.
This is pretty gross, but I could read this. Oh my gosh. I saw it. Yeah. All right. It says that the sperm of a man may be putrefied by itself in a sealed cucurbit for 40 days with the highest degree of putrefaction in a horse's womb. So they put it in a horse's womb, or at least so long that it comes to life and moves itself and stirs, which is easily observed. After this time, it will look somewhat like a man, but transparent without a body. If after this it be fed wisely with the arcanum of human blood, and be nourished for up to 40 weeks, and be kept in the even heat of the horse's womb, a living human child grows therefrom, with all its members of another child, which is born of a woman, but much smaller. So,
I mean, but that's just like theoretical, you know.
Right.
Right.
Oh, the horse? No, the horse thing can happen.
Absolutely.
I mean, but that's just theoretical. It said that it said something about the sperm being putrefied. I mean, that doesn't mean that the sperm by itself is growing into a human.
Yeah,
Right.
either
I
way, it's still not answering the original life, you know.
I guess that was
right.
claimed to be from Paracelsus. That was claimed to be from Paracelsus, who lived from 1493 to 1541, in De Natura Rerum from 1537. That's it was what it's just claimed of outlining, yeah, the how to create a homunculi.
I've
Wow.
heard that a lot of the reasoning behind this like alchemical homunculus thing comes from this belief system. I forget what it's called, but basically that everything is already in its form just on a small scale and that it grows. Like the idea that there is like a little tiny man in your sperm and that the woman is just a vessel for it to like grow within. And so you see how like that would kind of be like all we got to do is find a replacement for the woman's womb because there's already like a little tiny person inside of my sperm and I just need to find the right vessel to put it into that will also facilitate its growth into a larger size because it's already there on just a really microscopic scale that just needs to be
Do you believe
expanded.
that? Like, do you believe what you just described?
No, I don't believe what I just described. I'm saying
Okay.
that was there's a term for it, and I don't know the term, but that was the mindset that a lot of these techniques were based around. That there's a little tiny man inside of your sperm, and you just need to put him in sort of an incubator that will allow him to develop in size and grow larger, because his complete form is already there. But obviously, that's not how modern science or anything believes.
I guess I can see where they may have like thought that because they didn't know anything about the egg, and it's like, oh, you just put this stuff in here, and then a baby comes out, and it's like, okay, like, I mean, I don't know. I
Just
guess that's
putting dough into the oven.
yeah, huh. I know one thing that's really cool that is like interesting to look into is like what happens at fertilization. Because what happens is once a sperm gets into the egg, it opens up these ion channels and I mean, it's not called an action potential like in a neuron, but it like lights up. It like does the same thing that happens in an action potential when the cell, I think, moves these ions across the channels and stuff. Anyways, that's how the egg hardens its like membrane or whatever, so no other sperm get in there. It's like trying to only get one. And so and then I don't know, this process has been observed like under, different kinds of microscopes and stuff. And I've seen videos of it like lighting up and stuff, which is like so cool. I feel like it's like the light of life, it's like, the breath of life or something.
Yeah, does that
seen
happen?
that.
Does that happen in animals too? And then, like, to what degree does that happen even in insects? Do you know?
I don't know. That's a good question.
Yeah.
I found the term, by the way, real quick. Sorry. The term is called preformationism. The idea that sperm contained a little tiny person inside of it, and that all you need to do is put the little tiny curled up fetal position microscopic man into the right vessel to facilitate its growth into a full sized man. Preformationism.
That's interesting. My next block in school is reproduction. So this whole topic definitely gets me excited. So maybe I'll
for
come
sure.
back in 2 weeks and we'll talk about some details or something.
Amazing. I have one more quick question, Dan, if that's okay. I know you guys are busy. Thank you. Are you familiar with the lecture from some people, including Dr. Hovind, that try to outline the lies in the biology textbooks? Like one in particular was the slits. Yeah, the slits in the human embryonic stage, that seem to have been exposed as lies from George Hankel himself. Do you notice any misinformation in the biology or chemistry books?
Wait, was this guy Ken Hovind the guy who challenged? Was he Japanese? Maybe.
Oh no, Ken Hovind. He's from America. He's from the
Okay.
U.S.
He does
But-
answers in Genesis, I think.
The guy who does Answers in Genesis.
Yeah, they might be outlining those too. But I remember Kent Hovind himself. He had a lecture specifically outlining all the lies that he's found in the textbooks, and one of them was he was exposing what happened back in the 1800s. It was Hankel. He drew slits, gill slits on the human embryo, and it was found out to be that completely forged. I was just wondering if you guys have noticed any sort of misinformation.
Well, like I can tell you, like, so from a different perspective, this is also like from Jack Cruz talking about this Japanese scientist, and he spent his whole life like questioning the sodium potassium pump. And like if someone messes, like if there was something false before and then we build on it and build on it, build on it, then like we could get way off the track and they can have a whole line of false information. So like I don't know, I definitely am not going to like support the idea that the sodium potassium pump is false because it's like so essential. That's not, but there, and because you start like proving it with other things. Now that was kind of off topic to your question. Like I'm going to go look up this Ken Hovind person and learn more about what he said, but like I guess my point was how much information in our biology books are false. I mean, how, like, so I'll take you back to another quote that someone said was they were like, how much of our medical textbooks are false? And a lot of it might have to do with what's not that there's a lie there, but that there's so much stuff missing that's taken out. And let's say it's over 90% is what that person said. And I guess I'm going to refrain from saying that like there's so many lies in the biology book because what it's just observing like little bitty pieces of a whole part. I mean,
Yeah,
I
thank
think
you. I'll post the link to his lecture, and then at your own pace, you can find out whether or not it's true. I tried to look into the George Henkel himself. He was tried by peers from his university at the time because he was trying to show that the human embryo had gill slits, but it appears to have been faked by him and still in the textbooks. Yeah, but
yeah,
I'll post it to you there.
I wanted to talk about the gill slits. Yeah, so the gills, like if you're gonna, they're called pharyngeal arches. So you have 6 pharyngeal arches and corresponding clefts. Okay, and this is crazy, but like organs from your body develop from those clefts. There's pharyngeal arches, pouches, and clefts. And man, I gotta also study more on this. But like I know, for example, your third and fourth pharyngeal cleft become your inferior parathyroid gland and your thymus. And your thymus makes your T cells for your immune system, and your parathyroid. No, it's not fish gills. They might someone might think that say that it looks like it exactly, but it's they develop into different things. And like the fifth pharyngeal arch like breaks down. Your first one forms the bones of your face and your jaw. And so if you have and you can replicate this, if you were to like destroy the first pharyngeal arch, then an embryo, a baby would grow missing its jaw and stuff. I mean if you want a good example, look up the George's syndrome. Look up the George's syndrome, and that's when I learned about. It's like you don't form a thymus, a thyroid, um, and some other stuff like other things like the roof of your mouth, your palate doesn't form right. Okay.
Yeah, I've heard that. Thank you, Dan.
Let's go back to the biology book, for example. You mentioned things missing from the biology book. Like, which biology book are you talking about?
That wasn't really me that claimed that, but what I'm saying is that there might be. Well, I could really talk about this a whole lot. I mean, your medical books are not going to talk about herbal medicine, right? I mean, because of the Warren Commission, John D. Rockefeller funded the Warren Commission to go around the country and to make sure that all medical schools were teaching the curriculum that he wanted to be taught.
No. Okay.
Yep, Rockefeller medicine. Rockefeller medicine.
I agree.
So let's check this out. So as you know, as you progress through your education, right? There's another edition or more advanced edition of your text, right? So like for example, I have family who are in the medical field, right? So they took the biology track in high school. They took then they went on to junior college, right, where they studied like
Just
pre-med
get to it.
and so on and so forth. So your information gets more advanced as you get through the process. So there's nothing missing. What's missing would be things that are not yet said, like there's not yet a consensus to ensure that what's communicated to the students can be used or is in the field. You don't want to communicate things that are not totally verified and all these kind of things, right? So there's nothing missing from your textbooks. No,
Yeah,
if you're
I see
talking,
what you're saying.
now, if you're
Like
talking specifically about herbal medicine, a lot of regular pharmaceuticals is created from herbal medicines, but it is refined and to make sure it targets problems. When
Yep.
you're taking herbals, natural herbs, you have to be careful because they are not refined to do the job. So you have to read your labels. That's why the CDC really doesn't regulate. It's you have to do your own discernment on those things.
Yeah, I mean, sure, there's a whole lot to this discussion. We just opened up a
Right.
can of worms, but
Right.
what you like, about the I mean, sure, I think that you can get a whole lot more profit if we take this leaf from a plant instead of using in the leaf. We just isolate one chemical from it. We can patent it, and then we can develop it. And while it used to be a lot easier, now it costs about $1 billion to develop a new drug. Then we can charge people, say it was like Ozempic, for example. We can charge, which was found in the saliva of a lizard. We can charge them almost $1,000 a month for it, and they'll have to be on it for the rest of their life. I sure think that's a much better business plan than teaching people which plants that they can eat out of their yard. I mean, if that's what we're
Right.
going for,
You have to make sure you study the plant. properly. It's all of all of its properties to ensure you're not putting stuff into people that could harm them short term, mid term, or long term. Right? It's a
Well,
process.
Geo
Phase
taught
one,
me
phase
a
two, phase three
word.
styles. Right?
Have you ever heard of cofactors?
Yeah, cofactors are they're non-organic enzymes essentially. They're required for enzymes, but they're for it to be a cofactor by definition it's non-organic. And I don't mean as in
Oh, okay.
like doesn't come from nature or something. I mean as it's not an organic compound, which are ones that are made of a bunch of carbons and stuff. They're like metals. Like metals can be cofactors.
Oh, okay. I guess I meant it in a different way. Not that you're not completely correct in your understanding of its
Uh-huh.
definition. I was actually speaking to the other guy why there might be a lot more importance than he thinks to using herbs themselves and not some sort of super refined version of whatever particular
Oh yeah.
molecule that you think is like the active ingredient, because maybe these herbs are designed in such a brilliant way that they are best, utilized in their full delivery without removing anything or refining things, because the other, and this is the way I was using the word, the other cofactors involved with, say, THC and marijuana may not have as potent of an effect or a healing of a effect if the other things that are inside of the plant itself aren't there alongside it as utilize this herb. Maybe singling out one part of it can only do 60% of the benefit that if all the other ingredients that it contains were there alongside it to work with.
Yeah, yeah. I mean, you made a good analogy to cannabis. I mean, we know there's hundreds of cannabinoids, and it's like, oh, we're just gonna take, Delta 9 or something. What about all the other ones and everything else?
Right.
I mean, I guess another analogy like to health would say like, oh, I don't need to eat an orange. I took some, vitamin C pill. Well, it's like, well, it's
Exactly.
not, not gonna be absorbed and used the same way and stuff. But also, like, I guess I can argue for the other side, and we could say, well, if we know that this chemical here is going to target this protein and then perform this function, then we can just like I don't know, purify that, and it will be more strong of an effect. I mean, that's true to an extent, but yeah, I think you're right
Maybe.
about the importance of like consuming the raw food itself. I mean, as long as you know it's healthy, like, it's not dangerous or something.
Yeah, I'm sure there's science behind it, and when it comes to the level of understanding that we may or may not have, it may come down to a level of faith guiding your judgment to trust a more surgically, aimed, extracted specific molecule or whatever from this plant, or if you just believe that the plant itself was designed to be like a helpful tool to humans in the world. And
Yeah,
I
definitely.
just gut feeling trust the plant if it helps me, I'll take the whole thing, please, not just some sort
Yeah,
of chemically, what do you call it, distilled version of it.
yeah. I think that's smart.
Well, remember, scientists, researchers, these folks, they're doing what you're saying. You understand me? They're not just in labs cooking up something just because of a feeling or a whim or to harm the public. They're doing exactly what you're doing. They want to make the public healthy and keep the public healthy as possible. That is the motivation. Science is objective. This is not subjective. Primarily, it's objective.
Has science ever been hijacked?
Uh, uh, that's a loaded question, man. I mean, I think
Oh,
you know the answer to that.
Oh yeah, yeah. That was just taking it to an extreme to make a point.
That one's for Big Man Robert.
Oh, everything has been hijacked, even airplanes. But the point is that is that primarily,
Shout out to No
scientists
Planes.
are, that's why you have to get money out of the system. That's why you have to take money out of it, right? I don't want to turn things politics, but it's like having the billionaire on the Resolute desk. That's bad.
I mean, scientism has essentially always been after power, has it not?
And money.
No. Science is never primarily after
Okay.
power. They're after
Is science?
primarily
Would you
moving humanity forward. That's
would
their
you say that
primary goal.
science is under with the going forth with the endeavor to sort of expand knowledge?
Yes, expand and
Cool.
improve, make us
Yeah,
better.
and there's a famous
Knowledge.
quote by
Right.
the famous scientist Francis Bacon, and he said that knowledge is power. So science is trying
Yes,
to accumulate power.
knowledge is power. Just like so, going back to what the gentleman just said, him on his own, he wants to learn how to grow his food and make sure it's right. So he's gaining knowledge because knowledge makes him powerful. It's the same thing throughout the entire system. We're learning, it makes us more powerful, makes us be able to have dominion over our world. That
See,
is the
that's the
goal.
problem. Yeah,
Right.
exactly. It is to have dominion. Yeah,
Right.
that is
That's
kind
right.
of part of the issue. It's it's to have dominion over the earth,
Mhm.
and the individuals and the human beings also within it.
Yeah, God says do that. Remember, that's in Book of Genesis. You know,
Yeah,
we're
that's
doing
why
it.
I've got problems
We're
with
doing
Apple
it.
tonight.
It's the Book of Genesis. That's all it is. How are you going to have dominion if you don't try to understand it and study it and that's what scientists are doing. They're actually doing what God said we should do.
Yes, but we have to remain vigilant because science can be used against everybody. So that's why I believe we should have an updated version of the scientific method, including adding a code of ethics, because most people take it for granted that everyone's just going to have uh, do science, including uh, their own guide of ethics, meaning they're not going to falsify. But unfortunately, I think we live in a time where that happens. And my example would have been the doctors who were claiming that cigarettes had no link to cancer, even though it was uh, found out to be true, and they were just covering it up for many, many years.
Yeah, no, I agree with the code of ethics. Like, for example, stem cell research. Scientists want to grow certain limbs and they want to work, like duplicate, they're duplicating the sheep, so they're going to want to duplicate a man. That is why the government is stepping in and saying, hey, let us have regulations on certain behaviors, right? The government is the people, government is us. So that's why we have to elect people who will put those kind of guardrails in. You understand?
Yeah,
Yeah. I
good
think
thinking.
like from a more broad perspective, like just defining science, just being a systematic method of finding and utilizing knowledge to, then find more knowledge, and the hopes should be to benefit, all people in a true beneficial way. I mean, but science opens up door, or it gives you a key. It gives you a key, and you can decide which door you want to open up with that key. So you can either choose to do good things or bad things, and the motivations that make people do certain things are broad. But like I was just going to comment and just say that, in general though, I feel like I do trust the process of science, and most scientists are probably, doing their job trying to find something that's going to be helpful to humanity. But because of what I'm doing and stuff, do believe that the system can function, but right now it needs major adjustments. I mean, I can just tell you from personal, what I'm experiencing right now. They are not telling you about the dangers of gain of function research in medical school or in any big time, like research program or something like that. No one's talking about it. However, we all just had to live through an example of how gain of function research killed millions of people and shut down the world for a couple years or whatever.
Maybe.
Yeah.
Yeah, but that was the point.
Yeah, you think that's what they were trying to do? Yeah, you don't do it by accident.
Yeah, these beings aren't fucking around. They know exactly what they're doing. Kill humans.
I'm not sure I'm ready to believe in any origin story of this illness, this supposed illness.
Let's change the topic. Let's change the topic. If we're gonna keep talking, let's just talk about something else. Y'all come up with
Right on,
something else
right
to
on.
talk about. Anybody else have any comments about abiogenesis and stuff?
What were y'all talking about?
Well, they were talking about science stuff, and so I just mentioned generalized belief in science and the process of finding knowledge. However, science gives you a key, and you determine what door you open up with it. So you can either do good things or harmful things.
Yeah, weren't they trying to make a virus that bound more tightly to like the receptors in the lung cells? I mean, that's kind of what they were trying to do, but in order to like find vaccines or something. But they should keep it more heavily controlled if they're doing that. And I don't agree with doing that either. But yeah, and other scientists, even the White House admitted now that that's probably the most likely cause of what happened at COVID with gain of function research. So it's not like a taboo subject anymore. I think they finally concluded that that was most likely what happened. So.
Who from this community found you guys? I mean, the egg might be on my face, but I've been around and I don't think I've ever met you guys before. So like, someone here must have I don't know, met you and invited you to give a presentation over here. I was just wondering who was that?
Well,
Bridge
I
that
did
gap.
it on Cause first, and then I was sharing with them about some of the things I presented, and they said, "Yeah, you should come present over here." I can't remember who said it, but there was a couple people who said it in the space one day.
Totally.
And yeah, I don't remember who said I should present it
think
here,
that
but
was me.
somebody did. Well, I'm glad I did. Thank you.
Yeah, I don't see too much hope for abiogenesis outside of creation. I suspect that future theories are going to be pretty interesting and funny, and maybe even sad. But yeah. Thanks for. Oh, I did have a question. Where can we find? Is it possible to review your slides at all, just for reference in the future, or?
Yeah, I'm trying to find a way I can like make them public, without like the embedded information on there. I think
Oh,
Sam's
good
gonna
idea.
help with that.
Okay, thank you. I like
You can
the
DM
slides.
it, and then once I do that, I can try to give it to you.
Okay,
I've
no
got a
rush.
macro that prints out the slides. If that helps.
So you guys just dismantle Abiogenesis in the whole server? Is that what I'm hearing?
Yeah, we just totally destroyed it.
Good job, guys.
Yeah, thank you, Sam.
On the homunculus topic, Adam is the first golem by definition. Not to say it wasn't a very special golem, but it does fit the description. You guys know about that Jewish mysticism story that Frankenstein is based off of, or Frankenstein's monster is based off of?
Yeah, can you refresh our memory?
Oh, I'd love to. So there's this ancient Jewish story of like this Jewish mystic, right? And his town or his village is getting like attacked every once in a while by a neighboring village. And so he uses his, Yahweh endowed, powers, where he builds a statue of clay, a very large statuesque, man shaped, thing of clay. And then he inscribes this word on his forehead that, brings this, clay man to life, right? And it's a golem. That's like the definition of it. It's like clay statue brought to life. And then the whole way it relates to Frankenstein is that one day, or actually every night after he, brings it to life at night to guard his village, from whoever might try to attack the village at night. In the morning, he deactivates it by smudging the thing he engraved into its head. Erase the words he engraved onto its forehead. But then after doing this several times, one day his finger slips and only erases like the last letter in the word that brings it to life. And apparently the word like live versus the word attack are only a one letter difference, something like that, right?